Good morning, everyone, and welcome to this MSA UCL sponsored meeting 2025. We've had one before or two. Yeah. So this is the latest one in the series. I am Kailash Bhatia. I wear two hats today. I'm UCL consultant neurologist there, and a lot of my colleagues here today. So I'm really proud that we'll be sharing some of the work which we've been doing at UCL. But I'm also the chairman of the UK MSA Trust. I've been privileged and honored to do that for the last 3 years. And Karen, where's Karen? There she is, yes. And, under the tutelage of Karen. And Andy, he's here. Andy Barrack, he's right. And a lot of the staff at the trust. So we all welcome you to this meeting. As you can see that I'm really sort of delighted to see how much progress there has been in this field, which you can see reflected in the program which all of you have. When we used to talk about MSA some years ago, we would be struggling to talk about the diagnosis and talking about patient journey, all of which is, of course, still very important, but there has been so much progress on the research side. And this morning, we'll be hearing a lot about that. Viorica, who was our first fellow for the MSA Trust. And then others have followed as well, Dr. Go, and I gather Linda here too. So, and of course, a lot of work carried out in this area by Henry Hulden, Professor Henry Hulden, my colleague. And you're going to hear a lot of this morning from the UCL side. And the afternoon, of course, is devoted to our main patient-centered approaches for improving their care. And I must say that I have been actually in the last 3 years, while I've been there as the chairman of the MSA Trust, I have realized, which I didn't before, I must say, the amount of work which the MSA Trust UK does for the patients with MSA in this country. The support services, our MSA healthcare specialists who go to clinics, the various boards we have and specialist committees for the scientific panels, etc., forming the research for the future and also for our patients. A variety of things, voice banking, counseling services, etc. It's really amazing amount of work. And I think some of these people are here. You could meet them in the afternoon in the breakout sessions. Now, we made good weather for you, which is, you might have realized, but you can sort of enjoy that perhaps in the breaks, but you're going to be here because the program is really good. Some other sort of things to keep in mind. We are not expecting any fires or fire alarms. So if you hear a fire alarm, then please note where the fire exits are. Keep that in mind. And we're going to have the keynote talks in the morning, and then there's going to be a discussion session after each of these sessions. So in the first one, I think we are going to start this morning with Viorica Chalban chairing it, and you're ready? Excellent. So we won't waste much time and we can go on to the program. Viorica.
Thank you, Professor Bhatia. And it is a real pleasure to have everybody here in the room. And of course, for our keynote talks this morning, I'm really delighted we managed to bring over people who I really appreciate, but also who are really good friends of ours. So for the first talk, it's my pleasure to introduce Professor Andy Singleton, and who really is the leader in the neurodegeneration and neurogenetics field. Andy completed his BSc at the University of Sunderland and the PhD at Newcastle. And then he went on to study genetics of Alzheimer's disease, Lewy bodies, Parkinson's disease, and lead international and global consortiums. He has many accolades. Recently, he was awarded the 2024 Breakthrough Prize in Life Sciences, and for like the major work he's been doing in the field. And today, he will present about the Global Parkinson's Genetics Program, the GP2. This is a large international effort to improve our understanding of conditions like Parkinson's disease and related diseases. And importantly for us, for our community, MSA was recently included as part of the conditions in the Parkinson's Plus program. So it's my pleasure to invite Andy to tell us how we can actually work together with the program and globally with the network he's established.
Right. Thank you so much. I think this works. We'll see. Morning, everybody. So thank you so much for the invite to come and talk today about a program which I really love. I've been involved with this program since its inception back in about five years or so ago. Yeah, I'll tell you a little bit about it over the next 25 minutes. Yeah. Okay. All right. So the Global Parkinson's Genetics Program is an initiative that started about 5 years or so ago. It's supported by the Aligning Science Across Parkinson's initiative. And we work very closely with the Michael J. Fox Foundation in order to establish it. It started really in Parkinson's disease and focused on trying to understand the genetic basis of disease and to do so in a global context. So really expanding our analyses outside of the typical work, which is centered in Northern European and North American individuals, to work in underrepresented populations. A couple of years after we started, everything seemed to be going pretty well. So we expanded in a couple of ways. We expanded to include a lot more whole genome sequencing. I'll talk a little bit about that later. We expanded to include DLB, PSP, MSA, and Cook-Basal syndrome. We expanded our underrepresented minority work. We also did a lot more work in prodromal and at-risk individuals. I won't talk too much about that. We also have one program now which is focused on returning results to patients. So we're really a genetic discovery effort. So our work is research genetics that typically don't get returned to patients, but we sponsor a study called PD Generation, which has now returned genetic testing results to about 20,000 people. We are, the plan is for us to go through 2029. I think we'll probably end up going longer than 2029, but having this long runway for funding has been incredibly beneficial, particularly as we set up collaborations around the world. So the mission is a simple one: to really accelerate understanding and to do so in a global context. If we think of precision medicine, if we think of matching a therapeutic strategy to the mechanism that underlies disease in a patient, then obviously it's important to understand what that mechanism is in each patient. And we need to do so in populations around the world. So using genetics to understand disease around the world. The path is a little different from the typical genetics path. So if you were doing this 20 or 30 years ago, essentially what you would do is a well-funded lab would go to an underrepresented population, collect samples, usually with a local collaborator, take them back to their lab, analyze them, publish them, done, one shot deal. This is different in that the idea here is to build a global research community. So a large part of our effort, and this is really thanks to the funders, a large part of our effort is around building capacity in groups all over the world, so that the people that are analyzing the data are the groups that are collecting samples. So we're just trying to build this continued capacity so that even when GP2 goes away, there's the skills and the resources for those groups to continue working. Of course, the output of this is to create a discovery engine. Researchers all around the world generating new findings. It would be a failure if every paper came out of my lab or out of the very well-funded labs. I think that would just be the old system and we wouldn't be making much progress. A success here is where we're just generating ideas and answers all around the world. So there are four kind of pillars, I would say: global collaboration and capacity building. So again, building up these collaborations, building physical capacity. We actually build some physical resources in populations around the world, but building a lot of skill capacity around the world. One of the most fun bits has been growing the next generation of leaders. So you can imagine we have a fairly large leadership structure. There are a dozen or so working groups. There are more than 100 people involved in kind of the leadership structure. And for each of those working groups, there's a leader, someone who's a bit gray like me, who's in charge. But coupled with that is a new researcher, a junior researcher who works side by side with the leader. So every kind of aspect of the work is to find talent, find early talent, and grow them and give them opportunities to learn how to lead. Transformative genetic methods. So this is what you would expect. We're generating genome-wide genotyping on about 250,000 people. We're generating whole genome sequencing on just shy of 100,000 people. We have generated long-read sequencing on about 1,000 individuals, and that will continue to grow. And then the last piece is accelerating discovery by democratizing data. So the genetics community has been pretty good at sharing data for the last 10 years or so, but this is a bit more than sharing data. This is making data available, but also providing the compute capacity to be able to analyze that data and the skills and the training to be able to analyze that data. So you're really enabling research. So again, this comes back to this idea. If we have all of these things in place, we're generating skilled researchers around the world with the resources to be able to analyze their own data. This is how data and sample flow works. A PI joins the study, they send in DNA, they send in clinical data, we harmonize the clinical data, we generate genotyping and whole genome sequencing, we harmonize that. The first people to get that data are the investigators that sent the samples in. So the data immediately goes back to the investigator at a variety of levels. They can get summary reports, they can get raw data. The second phase is the data goes into the GP2 data and storage structure. So again, this isn't just a place that stores data, it has attached compute. So you don't need your own supercompute cluster, you log into ours and everything can be run there. It's all cloud-based. And then the last part is the research community gets access to all of these data. We use the AMP-PD platform as a way to access the data. So really anywhere where you are in the world, as long as you have a web connection, you can access. We have now 80,000 samples genotyped, 30,000 or so whole genome sequenced. You can log in and access that almost immediately. Building global research capacity. This has been a lot of fun. Of course, you want to be able to build the skill sets to analyze these data. So we've generated a whole list of things to enable that. The first there's an online learning system, around 70 or 80 courses available in 100 different languages, which really can take you from not knowing much about computers to all the way through to doing very complex genetic analysis. We have over 1,000 users of this, about 35% of which are from low and middle income countries. As well as building these virtual resources, we also build some physical resources. So we built a biobank in Lima, Peru. We're building one now in Lagos, Nigeria. And again, this is about enabling research in the local areas and giving like sustainable enabled research. Another way we teach is to go and hold courses. So particularly in data analysis. So you can see over the last 3 years or so, we've been really all over the place. And the structure of this is that we take 2 or 3 people from GP2 who are skilled in data analysis. They go to an area, teach a dozen or so individuals, identify someone in that group who's really great. The next time they go back, that person helps to lead the training. And then the third time, that person is doing the training by themselves. So it's training the trainers and trying to create this self-sustaining skill transfer. We sponsor quite a lot of students, so we have PhD students and master students around the world. There's actually another PhD call now. I think it closes May 5th. This is for PhD students registered in underrepresented populations, working on any aspect of the Global Parkinson's Genetics Program. One of the key things in this program was that the PhD students are registered in their home country. We really want to create skill sets and skilled people who are going to stay in their countries and help grow the skill set in that space. So what goes into GP2? What kind of cohorts do we have in GP2? They vary dramatically. Many are cross-sectional, many were collected, a decade ago, 20 years ago. We are, however, collecting a large number of cohorts that have very varied data. Some almost nothing, some immense amounts. I mean, if you take something like the PPMI study that has 20 years of longitudinal data, imaging, biomarker data, et cetera, we don't collect that data, but we collect metadata. So we have the genetic data at hand, we have clinical data at hand, and we know who has, for example, imaging. We know who has CSF. We know who has plasma. So if a member of the GP2 network says, I'd like to run a plasma biomarker, we can connect them with every cohort around the world that has that. We also support new collections. So this is something that's happening more and more. You can see we have new collections scattered around the world. There are some areas that we're still trying to target. We still need to grow programs in the Middle East, for instance. But this is growing. I'd say the good thing about these new collections is that they're fairly well harmonized, so that the same kinds of things are being collected in all of these spaces. So what does this give us? These dots on the map are the number of sites in each country. So we have around 70 unique sites, about 250 cohorts. There are 210,000 samples at some way fairly far through the pipeline for onboarding, although we're now projected to collect about 320,000 samples. There is data today available on about 75,000 individuals, genotype data on about 75,000 individuals, so you can log in and grab that. There'll be about another 20,000 in July. So we do releases every 6 months. So I think this is a good and useful resource. What kinds of things can you do with a resource like this? So I just want to give you 2 or 3 examples. Although we're 5 years in, that's still pretty early for a large effort like this. First couple of years are kind of spent operationalizing things. So we're really only 3 years into starting to generate large amounts of data and do analysis, but it's already bearing fruit. Multi-ancestry genome-wide association study. So essentially what this does is to look for genetic association in multiple different ancestral groups and look at the differences and similarities between those groups. This was run by a guy called Nacho Mata. And the take-home message here is that there's quite a lot of similarity across ancestral groups, but there are some very distinct differences in terms of genetic risk for disease. So these differences are key for a couple of reasons. First, they allow us to fine map, to really hone in on what the key gene is that's involved in a locus. Second, they're differences, right? They tell you that there is difference in the genetic architecture of disease between different populations. Another study, I think, that really shows the power of GP2 is this work, coupled with another paper. So one of the efforts that we try to do is train people how to analyze their data. We were involved in a genome-wide association study in Nigeria, Lagos, Nigeria. And as part of the training exercise, after the first 1,000 or so samples were genotyped, we decided to do a GWAS. You don't find things with 1,000 cases. So it was really just a training exercise that was run in NIH, at UCL, and in Lagos, Nigeria. So virtually online, people would analyze, they'd meet every week or so. And stunningly, during this training exercise, we saw this incredible hit over GBA. And to cut a long story short, what this is a new variant in GBA that's a risk factor for Parkinson's disease. Unlike previous glucose 3-peroxidase variants, this is a non-coding variant. And it's a variant that affects disease in a slightly different way in that it destroys a branch point site. So you get what looks like an apparent increase in RNA, but it's non-functional RNA, and that leads to a decrease in protein and a decrease in activity. So this was a really neat proof of principle. It's really the first major risk variant for Parkinson's disease identified in a sub-Saharan African population. It's a new mechanism in GBA. And given the interest in GBA as a therapeutic target, it identifies a completely new population for precision trials, one that would never have been included in precision trials. Multi-ancestry European meta-analysis. This work has just been finished 2 or 3 weeks ago. It's on bioRxives already. But this essentially shows you the power of large numbers. So looking at European ancestry groups, Northern European, Ashkenazi Jewish, Finnish, and Icelandic, and you get a lot of hits, right? You get something like 60 new hits by increasing the numbers just a small amount. And the reason we get so many new hits is because so much effort has gone into harmonizing and standardizing the data. So the data itself is very, very clean. You can then take those data and start to make inferences about biology. So if you overlay all of those hits, all of the genes underneath those hits, and you put them on top of expression data, if you're looking here at regional expression data, you can say, well, what are the tissues where these hits are particularly enriched? The genes underneath these hits are particularly enriched. And I'm kind of stunned by how clean and nice this data is. But here you can see that if you look at tons of tissues, peripheral tissues, brain tissues, the genes underneath these hits are enriched in brain tissues, not in other tissues. So Parkinson's disease is a brain disease. You can also do that on a single cell basis. You can look at which particular individual cell types are enriched for expression of genes underneath these peaks. So this gives you a cellular context for genetic risk. It allows you to say that in a general way, the genetic risk for Parkinson's disease is mediated through gene expression in these cell types. So no surprise here that you've got substantia nigra neurons, dopaminergic neurons as the most significant hits. GP2 has a ton of projects. The way that the program works is that there are a set of very core analyses. So, for example, this paper here is a core analysis, genome-wide association of PD. There are no authors for this. It's just GP2. So we have a whole series of 4 papers like this where there's no author. It's a consortium authorship, so everybody gets credit. Outside of those core programs, we have a project approval process where anyone within GP2 can propose a project. And there are a few things that are assessed when they propose that project. Is it fair? So if they're analyzing the African data, do they include African investigators? Are they including the right investigators? Is there a training component? That's another component of it. And do they have the skill sets to be able to analyze the data? That last point, if the answer is no, that doesn't mean it doesn't move forward. It means that we'll find people with the skill sets to be able to analyze the data. So anyone can propose anything. You can see here we have around 50 or so active projects right now, and they really are coming in thick and fast. So if you become a member of GP2, you get to access all of the data in GP2 and propose any project and ask for resources to be able to complete those projects. So what about beyond PD? I mentioned that we had about 2 years ago expanded into Parkinson's plus disorders. So there are a lot more Parkinson's disease samples available than Parkinson's plus samples available, but a huge effort by the cohort onboarding group, Hugh Morris is the lead of that, has led to us bringing in more and more cohorts in diseases like PSP and MSA. You can see we have studies around the world in Parkinson's plus. This is where we are now. So PSP, about 4,600 samples available, MSA, about 3,500 samples available, CBS, 1,200, DLB, 5,000. These numbers will go up. They're predicted by 2028. I really hope that actually that number is a lot larger than what I have here. We have the support to collect new samples, and we are starting to collect samples around the world. There's a rather large MSA collection in India and one starting in South America. So I think there's a huge opportunity here to bring this data in and make it useful for the community at large. So the take-home message, I think that diversity is the key word here. Ancestral diversity is important for understanding the basis of disease. The more different populations we have, the more we can actually understand about disease in any single population. It helps speed up discovery. It also gives us some level of parity, right? I mean, if we're thinking about precision medicine approaches and we'd like to treat this global disease in a global manner, we need to understand disease in different populations. So there's both a social and a scientific imperative. We're already, even in these early days, using that ancestral diversity to find new things and understand new processes in disease. And I think we're really going to accelerate over the next year or so. There are a huge number of, this is something I haven't really spoken about, there are a huge number of adjacent opportunities in GP2. So we're a genetics discovery effort, but even if you're not interested in genetics, there's a ton of other things that can be done. If you're particularly interested in imaging, we can connect you with all the folks that have imaging data. If you're interested in biologics, we can connect you with all of the folks that have biologic data. So I really see this as a nidus for collaboration, as a space for collaboration. I think the other diversity component is diversity in the context of diagnoses, particularly as we start to think in a more sophisticated way about pathology and co-pathology and how there is a continuum across many of these diseases. I think we need to be looking across our classic disease categories and look at ways that we can kind of break down that categorization a little bit and understand how diseases overlap. A critical component of that is having the same data produced across neurodegenerative diseases, and hence the focus for Parkinson's plus disorders. Breaking down the silos is absolutely key here, sharing the data in one place and making it available to groups around the world. So if you're interested in joining GP2, if you'd like to hear more about it, if you'd like to hear about the opportunities here, I'd love to hear from you. It's a genuinely enjoyable program and project. There's a lot of energy. As I said, there are well over 100 people in the various leadership roles. There are 2,000 or 3,000 people that are members of GP2. So it's a big and exciting consortium and lots of fun. And the more ideas we have, the more questions we can answer. So I'd love to hear from you. I'd love to hear if you're interested in joining or if you have ideas about things we could do.
I hope this works now. And we will have questions at the end for both speakers. And now it is my pleasure to introduce Professor Vasilij Messner. He's a highly respected neurologist who has an extensive experience in both clinical practice and research in movement disorders. He is based at the University Hospital in Bordeaux, and his work focuses on Parkinson's disease, but a lot of it on atypical Parkinsonian syndromes, including MSA. And he has collaborated with so many of us over the years, bringing both scientific input, but also his experience with working with actual MSA patients. And today I invited the listeners to tell us about distinct patterns of progression in MSA. And I must admit, this is a tough topic, and I gave him a tough job. And looking forward to hear from your experience and from your research and clinical work. Please welcome Vasilij Messner.
Thank you very much. Is this working? Yeah, can you? Thank you very much, Veronica, for the very kind introduction and the invitation. Yeah, challenging topic. You will see today, I mean, you can challenge me afterwards because I will share a lot of unpublished data. I've never shared in a presentation so much unpublished data. We discussed this yesterday with Veronica, just coming out of the analysis machine at the Bordeaux Population Health Center we're collaborating with, and so hopefully there's some interesting news for you. But I will do the little bit three parts. The first is more of an introduction, coming back to the UMSARS, which is the measurement of disease progression. Of course, you're well aware about all these studies that have looked at longitudinal measurements of UMSARS 1 and 2, but you will see that in recent years there have been efforts to better understand also specifically the progression of individual items and which are the items that contribute and which are the items most sensitive to change over time, which is relevant for designing a clinical trial. I will then come to the new diagnosis criteria released in 2022 and show you how these new diagnosis criteria have impact on the progression of UMSARS and survival. And you may be surprised to see the results. And then in the last part, I will spend 10, 15 minutes maybe to share with you these new results based on a latent class approach, where we used data of the French MSA cohort now, which is a cohort that was created in 2007 and has now more than 800 patients, clinical data of 800 patients in. And based on the clinical data, we have identified several classes, and then I will show you also how the additional ancillary biological, biofluid and imaging studies have enriched and go into the same direction as the clinical data. But before I start, just my disclosures unrelated to what I will talk about. Yeah, this is just one illustration. There have been publications from the North American study group, the European study group, and that shows you just the progression of UMSARS over time. There have been some factors, but that has not been entirely consistent being associated with the progression. Some studies have found that, for example, the MSAP phenotype is progressing more rapidly than the MSAC phenotype. We haven't seen this in our data. What we saw is that patients with MSAP, when they came first to our attention, had a little bit higher UMSARS scores, but the progression, the curve was then quite identical. A factor that seems to be really consistently identified as a factor having impact on the progression of UMSARS is early autonomic dysfunction, and that is illustrated here by this blue line. These are the patients who had early autonomic dysfunction. And then another observation, which is currently not so much discussed, the gender difference, potential gender difference. We know from other disorders, mainly from Alzheimer's disease, that women are usually, when they die, have a much higher disability. And this is exactly the same what we have seen in the French MSA cohort as illustrated here. You can see the progression of UMSARS scores for men and women. Initially, the scores are quite identical, but then women have a much faster progression, while overall survival is similar. That means that women are dying with a much higher disability compared to men. I think that's interesting, and we will see about some data Tom Fulton may share about his Exeneter trials. Their gender may also have an impact on how treatment works in Parkinson's disease and multiple system atrophy. Beyond these factors, it's really quite poor what we know about the factors driving the progression of UMSARS. We have just recently completed, I apologize for the low quality of this image, just completed a retrospective study including 250 patients with sleep studies. And we wanted to understand, I mean, that's not totally original because there is data about that, about the impact of stridor on survival in these patients. And of course, we could confirm that stridor is a factor of worse prognosis and survival. But interesting, what you can see is stridor has also an impact on the progression of the UMSARS score, illustrated here. This is the group with stridor. This is the group without stridor, while patients with sleep apnea, as defined by an index, hypopnea index above 15, had no impact. And interestingly enough here also, once you adjust for UMSARS to the stridor remains an independent factor of prognosis. So there's probably much more to learn from data and from the different cohorts around worldwide. The initial cohort studies, this is an illustration coming from the MSA study group published more than 10 years ago now. And these, the understanding of the progression of the UMSIS 1 and UMSIS 2 and UMSIS total to UMSIS scores has allowed then also to make some kind of sample size estimates. What is the number of intervals necessary, for example, to see a 30% difference over a year or 40% difference? And this is one of the first papers that has done this exercise, bearing in mind that here patients were included that who are not necessarily eligible for a clinical trial because there were a substantial amount of patients already very advanced in the disease. And of course, that may not necessarily represent what the situation is in patients in early disease, which are more likely to be recruited into a new protection trial in MSA. Following that path, Horatio's group with the North American Synucleinopathy Consortium has looked at the progression of individual UMSIS items, and this is a very busy slide here, but you can see all the 24 UMSIS 1 and 2 items, and it has looked at annual change over time, and it has looked at the standardized effect size, and you can see that there are items that are progressing more quickly, and the good news for I would say clinical trial design is that most are in line with functional impairment, dressing here, hygiene, cutting food, utensils, using utensils, handwriting, walking, speech, gait. So all these items that are really relevant from the patient perspective, which we have learned also afterwards, keeping in mind that the initial development of the UMSIS did not include any patient voice. So that's rather good news. And based on these observations, Horatio's group suggested to use an abbreviated modified 11-item UMSIS for clinical trials. We have also looked at the level of the French cohort to the progression of the items, and you can see here the individual progression of the items of the UMSIS 1, the UMSIS 2. What is interesting here is to see, for example, the orthostatic symptoms, which is really very relevant. We all know that for MSA, that the progression of this item is quite poor over time, which means that this item, as it is in the current UMSIS version, does not really capture what the patient feels and what the severity of orthostatic hypotension. The same is true for bowel function. So that really illustrates that in the current version of the UMSIS, autonomic dysfunction is probably not very well captured. Same is true for some of the motor items. This is the UMSIS 2, and you can see here the resting tremor, for example. We know that resting tremor is present in 20 to 30% of patients, but not more, and that is very nicely reflected here, that resting tremor is probably not a very relevant item in the UMSIS. We have then also done the exercise to align this progression to different disability stages as defined by UMSIS 4. So UMSIS 4 score of 1, 2, 3, 4, 5, and you can see how over the general overall disability, the different items progress, and that has allowed to generate substantial information together with the North American group to better understand individual progression. Another information we got out of that is that, in fact, the contribution of the items illustrated here by the different colors is quite similar across disease stages, at least until stage 4 of the UMSIS 4, which means that if you're planning a trial in a more advanced population, the progression of UMSIS seems to be quite, or the contribution, the weight of the different items seems to be quite similar over time. So despite all these, I would say, really this very nice better understanding over the last 5 years of the progression of the UMSIS, we know there is space for improvement. I already mentioned the limitation that the initial development did not include any patient voice. We are also aware that, for example, some of the regulators, especially the FDA, is no longer happy, especially with the UMSIS 2, and has asked industry sponsors to modify the UMSIS mainly based on items of the UMSIS 1, which has led to several UMSIS versions now circulating, and that all this together came to point to say, well, I think it's time to work on a revision, and this is why the MDS convened a task force. I'm sharing together with Wolfgang Singer from Mayo, and we have made substantial progress over the last 3 years and are currently drafting the first version of the new UMSIS, and hopefully this new UMSIS will help us to fill the different gaps, and it involves also patient involvement, and we will see how it goes over the next, I would say, 1 to 2 years. We should be able to provide the new version of the UMSIS. I mentioned already that now we have several versions of the UMSIS, which is a little bit tricky, of course, and we have done some sample size estimates based on these different UMSIS versions. And as you can see here, this is the abbreviated modified UMSIS based on 10 items that was used for the Biohaven trial. That was a large phase 3 trial involving 340 individuals. And based on our sample size estimates, you would need, for a classical comparison of the change over time of the UMSIS, 136 intervals. And if you're using all data available over the trial, all visits, all 5, for example, 5 visits over a year, baseline, 3 months, 6 months, 9 months, 12 months, you would need only 100 intervals. When you look at the 11 items UMSIS, then it would be 118, so quite similar. 86 when you use the different data points for the patients, and still the total UMSIS seems to be the most sensitive in its own. And what would be very interesting, I mean, we have people here in the room who are developing the mascot trial, and I mean we have seen now a schizophrenic situation where we have a trial, a phase 3 trial, an MSA, with 2 different primary endpoints depending on the region. One satisfying the FDA, which is the modified UMSIS, and one satisfying European regulations, which is the total UMSIS score. So we will see how that goes. But I really, that stresses the need to come up with a UMSIS that is acceptable and agreeable for everybody, clinicians, scientists, and also regulators. So what about the clinical diagnosis criteria? I think most of you know that we have new diagnosis criteria that were released in 2022. What was the reason behind? The reason behind was that we were aware that the former category of possible MSA was not so sensitive in early disease, and also having in mind the recruitment into clinical trials in an area where the number of patients is limited, it would really be very important to increase the sensitivity in these early disease stages. That was one reason why the revision was undertaken. And the second is the category of probable MSA had quite high specificity compared to the post-mortem gold standard, but still it was not 100%. So there were two objectives of this revision. The first, to increase the sensitivity in early disease stages, and the second, to make sure that once the patient has at least a diagnosis of probable MSA or the comparable category, that then the post-mortem examination should confirm the diagnosis. And I think it's really relevant because when we're talking, for example, about biomarker development, biofluids, or imaging, of course, the obvious gold standard would be the post-mortem confirmation that indeed this patient had MSA, but it is absolutely an illusion to believe that we will be able to establish large cohorts where all patients will have post-mortem confirmation. So we really need a clinical category that comes very close to that state. And this is why these different categories were designed, clinically established MSA, with the idea to really be more closely to the gold standard and the clinical probable, with the hope to have a higher sensitivity. And then, of course, inspired by the Parkinson's disease field, where really the tremendous characterization of early disease stages or prodromal stages has been done over the last 15 years. The idea that in MSA also we would need to start working on factors that allow us to identify patients very early in the disease. So this is the table of the 2 categories clinically established. The difference to former criteria is that you need an MRI marker, an MRI abnormality, and probably that is what brings this category closer to the post-mortem confirmation. And then in clinical probable, quite different in a way that you can have a patient with clinical probable MSA without autonomic dysfunction. And I think that's from a concept is not so obvious, and we will see what that means in terms of specificity in the long run. The good news is there have been a few post-mortem exercise studies similar to what has been done in previous iterations, where brain bank data were challenged to clinical data. And as you can see, this is data from the UK brain bank, that the new clinical probable and clinical established criteria have quite high specificity already from the beginning. So once the diagnosis is suspected, it seems that the specificity compared to postmortem confirmation is quite high. while the sensitivity unfortunately remains, at least in that exercise, quite low. So not sure that in fact the revision of the diagnosis criteria has really helped or will help increase the identification of patients in early disease stages. But what is the meaning in terms of the progression of UMSAs and survival? Very busy slide. What you can see on the upper panel is are the new diagnosis criteria. This is for UMSAs 1, this is for UMSAs 2. This on the left is MSA-P, MSA-C, here again MSA-P and C. And the bottom is the former diagnosis criteria based on probable and possible MSA. And what you can see here is that at least in the first visit, when patients are coming first time to the different sites of the French reference center, the UMSA scores seems to be quite similar for patients with MSA-P now with this new category, clinically established and clinical probable, and the same seems to count for patients with MSA-C. So there's no real difference. It seems to be no, at least in these data, no real difference between the two categories. And also when you look at the baseline score, it's around 22 here. It's maybe a little bit lower. This is what we have seen before in MSA-C patients. While in the previous categories, there was really a difference between probable and possible MSA with patients with a possible diagnosis, usually having lower scores, probably because the full-blown clinical picture is not present at the time. And the same is true for UMSAs 2. As you can see, quite an overlap for the two categories for MSA-P, similarly for MSA-C, while there was really a separation for the previous categories. In terms of survival, it's a little bit the same situation. So you can see here is clinically established, clinical probable. This is really the first visit when the patient first comes to the center, and you can see here what we also already observed with the previous criteria is that the median survival is around 3 years. In both groups is 3.1 and 3.8. There is a still small difference, but especially in this very early, I would say early time after the diagnosis, where the patient is likely to be enrolled in a clinical trial, it seems to be quite similar, and which is very different from what we have seen with the previous category, where patients with possible MSA really had much better prognosis compared to those with probable MSA. So the last part will now be devoted to this latent class approach. Latent class approach means to try to get out of the whole bunch of clinical data, subgroups following specific criteria and based on specific variables. And what we have done in this first iteration is to use available UMSAs 1, UMSAs 2, UMSAs 3, so the blood pressure measurements here in upright position, in supine position, and survival. And of course, the longitudinal measurements as well, this longitudinal component, to see if these data would allow us to identify different groups. And the answer is, with that approach, yes. There have been two results, either having 3 groups, 3 classes, or 4 classes. The 4-class approach brings 1 class with really very tiny numbers of patients, and this is why we decided to go for the 3-class approach. And as you can see is that the progression of UMSAs 1 is quite different across the groups. Class 1 and Class 2 looks quite similar in terms of slope, while Class 2, which is almost 50% of the patients, has a low progression, UMSAs 1 and UMSAs 2, and has also a better prognosis in terms of survival. The curves of the first and second class look quite similar. The only difference is that patients in this 3rd class, they're coming much earlier to us, already within 3 or 4 years, which means that these have probably a very aggressive form of MSA, and which explains why the survival is reduced compared especially to the Class 2. We were then wondering, what are associated factors, and this is a preliminary analysis showing that a diagnosis, that where we used here still the former diagnosis criteria, that a diagnosis of possible MSA was more frequently encountered in those with this second, more benign class. And these patients also had less frequently early autonomic dysfunction, so that comes a little back to what I said initially, that early autonomic dysfunction seems really to be a driver of worse prognosis. The years since symptom onset were different, and this is what I already mentioned, that in those individuals in class 3, they really came very early to the center, probably during their journey, their disease journey. The neurologist, the private neurologist who usually sees first these patients, has noticed red flag signs and then referred the patients. And that was different for class 1, where the mean disease duration was 4 years, and even more in this latent class, where sometimes the diagnosis is only made after 5 or 6 years. The age was similar. What is also noteworthy is that it's not very detailed for the time being, but just here the score of the MSA quality of life questionnaire, and you can see is that at the time of the first visit, the impairment in quality of life is similar across the 3 groups, and the same occurred also for the cognitive assessment. Of course, we're currently looking more deeper into this currently to see what other factors could be associated. There was no effect of gender, no effect of MSA subtype, so no impact of being MSAP or MSAC subtype, and the presence, the onset of the disease with either ataxia or Parkinsonian syndrome had no impact on the classes. We know from many studies now, it's really a growing body of studies about biofluids and imaging, and that was a very nice initiative led by Viorica a couple of years ago that brought together, I think, 200 samples of 200 patients, international collaboration, where we could see that there is a correlation between NFL, neurofilament light chain plasma levels, and the UMSS at baseline, and also an impact on survival. I mean that there was in the final version of the paper there was a little mistake, so it should be the opposite. There was a confusion of the colors. So this is the group with a higher concentration would have shorter survival, and that is really now consistently there are coming up studies illustrating that. And we have very recently run at the French level a cohort, the ASPIRE cohort. These are 40 patients corresponding to a clinical trial population, so very early in the disease, and these patients were seen baseline 6 months, 12 months, had MRI, DAT-SPECT, G3 trial points, clinical assessments, and also biofluid sampling and assessments. And what you can see is that in this clinical trial population as well, very clearly those who have the highest concentrations of neurofilament light chain are those who are diseasing earlier compared to those with the lower concentrations. So we're wondering if biomarkers, if that kind of biomarker would also be somehow connected, associated to the different classes. And as you would expect, at least for CSF, neurofilament light chain concentration, this is illustrated here, that the group, the class 3, with really the earliest manifestations and the shortest survival and the fastest progression in MSA scores, showed the highest concentrations of neurofilament light chain, lower in class 2 and intermediate in class 1, and no difference in total alpha-synuclein, no difference in total tau, no difference in GFAP across the groups, but we're currently expanding these data also to see if other biological factors may drive these different classes. Second question then, biofluids, okay, neurofilament light chain. Is there any change in imaging? This is just a very busy slide from the ASPIRE cohort, where we looked at brain imaging, progression of brain atrophy after 6 months and 12 months. And what you can see here is when we look, for example, to the stratum, that after 6 months, you see already compared to a control group and also baseline progression in the total cohort, and that then progresses over 12 months. There were differences between MSAC and MSAP, which is also relevant when it comes to clinical trials and using, for example, pons atrophy or putamen atrophy as a secondary exploratory outcome. But very clearly, that illustrates what we know from the literature now. Really a robust literature available showing that there is atrophy in specific regions involved in the neurodegenerative process. And we here see that these changes can be seen already after 6 months in this patient population. So we're wondering if brain atrophy would also be associated with these different groups. And as illustrated here, you can see is that the patients again in class 3 had the highest atrophy, the strongest brain atrophy in the putamen compared to class 2, and again class 1 was an intermediate. And surprisingly, there was no difference in the pons. So we really need to look here also into this. Why? What are the baseline atrophy values? does these baseline atrophy values play? We will also now enrich with progression of atrophy, because we have in a relevant number of patients at least a second MRI over time. So that will be enriched now. We will see how we could get more information out of that. Okay, and that brings me to the end. I think in the three parts, first part, recent studies have expanded our understanding of disease progression by describing really specific trajectories of individual UMSA's items and their contribution across disease stages. Second was devoted to the clinically established and clinically probable MSA criteria, which seem to be less distinct, at least in terms of UMSA's progression and survival. And we see what the impact will be on clinical trials with that observation. And the most recent, very recent, just recent results over the last few weeks, with the identification of 3 latent classes with distinct disease progression, biomarker profile, and survival. And of course, further enrichment hopefully will pave the way to personalized prognostication, of course, mainly for probably clinical trials, but I think it could also have relevant implications for clinical guidance of patients and caregivers, especially when it comes to difficult, challenging ethical questions such as gastrostomy or tracheostomy, where you would like to know what is really the prognosis of the patient if you offer that kind of invasive strategy. Thank you very much for your attention.
Thank you. Thank you very much for both talks. I would now like to invite both of our speakers, Professor Bhatia, and open up the floor for discussions. Question from Henry.
Impressive program over the years and the years to come. What are the 2 things, apart from genetics, you want to deliver over the next 5 to 10 years? Genetics, great in PD. MSA is coming with Viorica's help, hopefully. And but what else? You know, what 2 things, please?
Yeah. I'm guessing you're asking about scientific things.
Personal.
Yeah. Well, so
Growing. What can I deliver, or we deliver for you? You'll give us money
Mm-hmm.
to do, and we'll be.
Yeah, I'm gonna
successful.
I'm gonna slightly ignore your question and give you the answer that I want to give out. Building a robust research community that is not centered in one place. That's really important to me. So I'd like to deliver that. I think more broadly, having researchers who are not geneticists access the GP2 network to ask questions that they're really interested in. So using the structure that we have to ask lots of diverse questions. You can imagine also, and many of us are thinking about this, using the kind of network that we have to create precision trial ready cohorts is a potential interest, particularly as we start to think about running trials outside of Europe and outside of North America. And those are the key things. I would say one of the scientific questions I'm really interested in now is the genetic basis of SAA positivity, and whether that can put our genes into drivers of synuclein or response to synuclein. So I think that's it.
Maybe I can sort of, again to you, Andy, with this big resource and a huge amount of genetic work has gone into it, do you think that the days of monogenic discovery, or monogenic gene discovery in Parkinson's is sort of limited, or, you know, both in the 100,000 genomes and as best as I know in the GP2, so far, at least apart from the risk genes, which, you know, the Nigerian population and others, we haven't come across in the 100,000 genomes many new monogenic genes. And I don't think that in the GP2 either, so far at least, that we've come across that. And I mean, obviously it's ongoing work. The other important thing, I think, is how do we sort of deal with the differences in the let's say, environmental factors, the epigenetics, etc., and these different populations. And to my mind, that would be a sort of a major thing which perhaps GP2 could look into. So, the pollution in India is very different from the pollution here, and how is that contributing to the degenerative conditions such as Parkinson's or perhaps even MSA and so on?
Yeah, both great questions. So what we are finding, I think, are monogenic genes that are extremely rare drivers of Parkinson's and related disorders. I think the other thing that GP2 provides is there have been a dozen or so monogenic genes for PD reported in the last 4 years. As far as I know, only one is right. So we're able to establish what's right and what's wrong because we have a very large resource set to check things out in. In terms of environment, we just established an environment interest group. We have standardized environmental questionnaires across the new cohorts. I don't think that's enough, frankly. As you know, environment is a really tough nut to crack, but this seems like an obvious place for GP2 to go. Yeah. But it's not a cheap experiment, to do well.
More money?
Yeah, need more money.
Any other questions? Yes.
How you have shown us the different profiles. How would this understanding change drug development? So because these three profiles are showing different speed of progression, which is the primary endpoint in many of the drug development studies. So question number one. And question number two, do you speculate or do you know if drug response is different between those three profiles?
So I think, well, the first, it depends really on what you would like to achieve in your trial. If you're looking, for example, for a population that is the fastest in disease progression because you would like to have a trial duration which is short and you're very confident about the power of your drug, then you may want to have that kind of very rapidly progressive population. And it seems in terms of the progression less heterogeneous than this class 2. When it comes to the biology, of course, this is now where we will continue the biological enrichment to understand if there's specific targets that may be worth to be addressed in these specific three classes. And maybe finally we will come up with them going out of these three classes, even when you're enriching with additional data, to come with a more personalized prognostication where you can really track almost individual trajectories with a certain probability. But that's the beginning of the story. Of course, we now need to enrich, and we have already some ideas with Viorica how we can tackle that.
And I would like just to make as a comment, I really like this panel because Vasilios has presented so much data on the clinical progression, and we have the GP2 programs where the large genetic data can be integrated into informing like the missing pieces, between the two aspects. So yeah.
So the class 3 early autonomic strider fast progression neurofilament, yes. Is there anything with regards to synuclein in your biomarkers which might help predict progression and so on?
So
we looked at that.
No, for the time being, I just showed you the total alpha-synuclein concentrations in CSF, and that is the same. But of course, we would be very interested now to look at other species. We have some data, but not for all the subjects, phosphorylated and some oligomeric data. We have had a collaboration with OMA in that regard. So we'll have a look on those. But I think it's very fragile. We would need to really expand and use available biosamples because we have biosamples available. It's not all, the entire cohort is now 850 patients, and we have imaging for around currently 200 patients and biofluids for 150. So it is a subset of the patients, but still we have available biosamples, and we are happy to collaborate and to learn more about that.
Have you looked at the seeding assay in your
So
CFA?
not yet, but we have a grant from the Fox Foundation to do it. And the current challenge is, as always, my administration to get the contract signed, but we are very, very close. So I'm very optimistic that we will send the samples over the next few months to Amprion and get results back. And what would also be very interesting to see is not only these SA plus yes, no, because we had already collaboration with Laura Parkin in Oxford. She was also she's also working on a SAA. And what we could see in our very preliminary analysis based on 24 MSA cases that the kinetics the kinetics are correlated with the progression of UMSIS. So there's probably more in than just yes, no. And I think we will learn later about the clinical trial, the iterative trial, and we will maybe also learn about the challenging interpretation of type 1 and type 2 synucleinopathy based on clinical data, because in the alterity trial, we have included a couple of patients, and one where really I have no doubt that that is an MSA patient, clinically established MRI abnormalities, has type 1 synucleinopathy SAA. So
those kinetics data are really interesting, right? And they would suggest there's a different species. There's a different species here. We just don't know.
I think I don't know if you have seen this. Just came out a paper from colleagues in Auckland and Glenda Halliday, a brain paper where they looked at different on postmortem brain tissue on alpha-synuclein in neurons and in the glia. And what comes up, what has already been published, a couple of papers, that it seems that the neuronal synucleinopathy is so abundant in MSA. And what was really amazing to see in that paper is that when they used then proteinase K as an enzyme to digest alpha-synuclein from neurons and glial cells, they found that the alpha-synuclein in the neurons is much more resistant to proteinase K than in glial cells. And that may be then also connect to work from Michel Goddard and Carlos Soto, telling us that in MSA you have two types of fibrils. Are these the two types of fibrils? We will see.
The last one. If you not necessarily to have that scan at the time, but then later on develop very clear MS P. Is that misdiagnosis in such cases, or is it the gene that change later on?
No, we have seen, and we have published a small series, and there are others who have done this as well. There are some patients who really can go up to 15 or 20 years and can resemble Parkinson's disease for 10 years. We had a small series of patients who underwent deep brain stimulation, and then once the electrodes were placed, really the full-blown picture of MSA occurred, and all these patients had postmortem confirmation of MSA. But they had dopa responsiveness, they had fluctuations, they had typical topography of dyskinesia. You can see some of these patients, and we looked then also at the brains and did some kind of exploratory analysis. And the only difference we found in terms of new pathological markers was that these patients with these more benign forms had more neuronal alpha-synuclein in the stratum. But well, I don't know what that is telling us.
I just want to, there is similar data from the Queen Square Brain Bank where people who are pathologically confirmed and say they did have a PD clinical picture. So in life, they were diagnosed with Parkinson's disease, and there was nothing to suggest there weren't Parkinson's disease until the pathology confirmation. So I would not say wrong. They fit the diagnosis of Parkinson's at that stage.
One of the things about dyskinesia, do they get classical dyskinesia in this group? I don't think people have looked at that.
They did. I mean, I remember very well one patient. No, she was seen first by Pierre Pollack.
Well, maybe we need some better markers in that context. Okay, I think that's.
Thank you very much, both speakers.
Thanks.
So good morning, please. Good morning. So it's a pleasure for me to be here. So I'm Valeria Giudice. I'm a consultant neurologist and head of the autonomic department here in Square, and I'm grateful that I have been invited to chair this session. We're going to move, I mean, is a nice flow from Professor Meisner actually talk. So we have seen that autonomic is an essential feature in our patient with multiple system atrophy. And there are three important concepts really that affect, as we've seen, survival, then affect quality of life in patient is an essential criteria. But most importantly, we have solid knowledge now to see that actually in the premotor phase, so in the early stage of the disease, patient they can present with no motor features. So a third of the patient they will present to us with autonomic dysfunction. And this is a window of opportunity for us, thinking about pipeline for modifying treatment where actually we can change the trajectory of this pathology. So we have two international leaders as a speaker in this session. So we will start with Professor Horatio Kaufman. Then is Alex Roll, the professor in neurology, director of the Autonomic Center at NYU in New York, chief editor of Clinical Autonomic Research, and I could go on and on and on for a very long time. Has contributed immensely to the field of neurodegeneration and autonomic failure, and it's a pleasure for me to actually, the floor is yours, and it will introduce the concept of prodromal phase and what it means actually for our patient.
Can you hear me? This is working, yes. So, good morning. It's always a pleasure to be here. I was going to say that it always gives me goosebumps to come to Queen Square, but this is not Queen Square. It's Great Ormond. So I found out that I had to change my talk. So, as you know, Queen Square is the cradle of neurology, and for the rest of the world, it's always a place to aspirational and to emulate. And I still remember the first time I spoke at rounds here with Andrew Lees, something like 2,000 years ago. But again, it's a big pleasure. So thank you very much to the organizers, for Eureka, Henry and Valeria and Kailash for inviting me here. Both Andrew and Vasilius are a tough act to follow. I'm going to come back to the clinic and with a very simple presentation. And I apologize for my slides, but they're very simple. I have to tell you that I have to do them myself. So if you don't like them, you let me know. So you're all aware, you've seen these different variations of this graph. As you heard, the up to 50%, if not more, of patients with multiple system atrophy first consult a doctor for symptoms that are non-motor, right? So like other neurodegenerative diseases, multiple system atrophy, we know now, has a preclinical stage with only biomarkers, then a clinical state, which is premotor, no motor symptoms, where the symptoms are either autonomic or sleep. And then after that is the onset of motor symptoms with the overt Parkinsonian or cerebellar signs. Of course, this is in general, not all patients follow this, but most of them do that. Now, the time of diagnosis, as you know, is a few years after the onset of motor symptoms, because the diagnostic criteria establish that. So the big question is, can we move the diagnosis in this graph to the left? Can we make it earlier? And I believe the answer is yes. And that's why in the last diagnostic criteria, the modification, as you saw, what Vasilius showed us, there's a new criteria that nobody wants to touch. And in fact, it was a big argument to get it there, which is still an experimental or a research criteria rather than experimental. And that is possible prodromal MSA, which is at the bottom of the diagnostic circle. So what is possible prodromal MSA? Very simple. Is a non-motor feature, either neurogenic orthostatic hypotension, what we call pudendromic failure, or urogenital failure. You hear more about that with Jalish. Urogenital failure is erectile dysfunction plus difficulty in urination, particularly residual, increased residual urine. And the third one is REM behavior disorder, so either autonomic or sleep, plus subtle motor signs of Parkinsonism or ataxia. Of course, what are subtle motor signs? It's a big discussion. So, sorry. So, it's so the issue is perfect. What are subtle motor signs? Subtle motor signs are, you've all seen them, and once you saw them, you cannot unsee them, which are reduced arm swing, the same as in Parkinson's, usually on one side, slightly wider base gait, very small. When you see some air between the legs when somebody is walking, paucity of movement with bradykinesia, mask faces, mild incoordination, almost clumsiness, so mild that patients are fully unaware of the change. You know, we see these changes, but they don't complain about that. So in the definition, we said, okay, these are signs that in the opinion of a movement disorder expert, you know what that is, do not qualify as Parkinson's or cerebellar attacks. So here, let me go to the video tape. A 62-year-old female comes to the center. She tells us that at age 58, she started with lightheadedness and blurry vision, particularly when standing up or climbing stairs. 2 years later, the husband realizes that she talks and moves while sleeping, suggesting REM behavior disorder. At age 61, she starts with increased urinary frequency and sensation of incomplete bladder empty. We see her for the first time at age 62, and as you see in this graph, this is the tilt table result. This is systolic and diastolic. In red, you see heart rate, big fall in blood pressure, little increase in heart rate. The Valsalva maneuver that you see here has no phase 4. So clearly, this is autonomic failure. And in addition to that, the ratio between the increase in heart rate and the fall in systolic pressure, which is an indication of neurogenic OH, is very low. It's below 0.5. So it's only 0.2 beats per minute that the heart rate increases 4 millimeters of mercury. The systolic goes down. Very abnormal neurogenic orthostatic hypotension. We do a bladder ultrasound. She has 140 cc's, very high, over 100 of residual urine, clearly a neurogenic bladder. And this is what I'm probably going to focus in the next few minutes. She has a smell test and she has normosmia. You see here, these are, you scratch these things, you smell it almost like a lottery ticket, but you smell it and you can grade olfaction. She had normal olfaction. Again, 62, this is an exam. You tell me what you think, right? These are the movements. It's pretty good. We argue in the office, is this normal? Is it not? You should watch it and show the walking. Bear with me, look at that gait. You tell me, interested in the movement disorders expert, what do you think of that gait? Most people thought was completely normal. You know, I can show, sorry. I can show you that again, just once more. If you pay a little attention, you'll see that her feet are not going one in front of the other, right? Clearly, this woman is very thin, but you see here when she's walking, you see space between the legs. The feet are not going one in front of the other as a normal gait. Still, there was argument, is this a variation of normal or is it not? So here she got a diagnosis of pure autonomic failure and there was argument. 2 years later, this is what you see. Okay, so let me show her walking. I think is the ataxia now is evident and the white base gait, I think is quite evident only 2 years later. Right? So I'm gonna quickly, am I convincing you with the gait? So I go to the next. 2 years later, we have to go to her house to film because she's bedbound. And I think there's absolutely no argument how this has quickly evolved into. But you can see her walking. I'm going to. A year later, age 68, this baby died, and the autopsy report was classic multiple system atrophy. So, the clinical summary, as you see, she started with first symptom at age 58, just orthostatic hypotension at 58. 10 years later, she's dead. And where did we make the diagnosis? Here, when she had an overt Parkinsonian syndrome. I'm pretty certain that with the new diagnostic criteria and with awareness of those signs, we can easily make the diagnosis at least 2 years earlier, which of course is very important if we want a window for treatment. So how common is this phenoconversion from, and we call it phenoconversion because it's the same disease, right? It's not a new disease. That's why we use the name phenoconversion. How common is this from pure autonomic failure to manifest synucleinopathy? Well, we had this natural history study of the synucleinopathies, a study that went on and it continues going on for 14 years now with 8 sites. At the peak, there were 20 sites around the world enrolling patients. Now, the part of the data I'm going to show you is because here we enrolled at that time that everybody thought we were a little crazy. We were enrolling patients at premotor stage. So at every stage of the synucleinopathies or possible synucleinopathy, we were enrolling patients. Here at the premotor, either in pure autonomic failure or REM behavior disorder, right? And also they could enter here, but this is another part of the study. Now, in that first study, at least in the 10-year iteration, when we analyzed the data, around 1/3 of patients that presented with pure autonomic failure developed within 10 years a synucleinopathy. Now, as you see here, if you look at the lower part, most of those were Parkinson's disease, meaning 77%, 2/3 were either Parkinson's disease, but some, 23% were multiple system atrophy. So, this shows the Kaplan-Meier curves of that paper, yes, last year. And it shows in red multiple system atrophy. In the first blue is Parkinson's disease or dementia with Lewy body, and the cumulative effect in the lower, right? So you see a small amount, which is 1/3 multiple system atrophy, the other 2/3 are Lewy body diseases. So, and as you see, these are other, the two on the top, which is the same study, 2 iterations, is prospective, the other 2 are retrospective, but the percent of phenoconversion is very similar in every study, and the percent to MSA is also very similar in every study, a little lower in ours because it's prospective, retrospective is easier to get. So it is clear that pure autonomic failure is a prodromal synucleinopathy. I think we all agree on that. Now, how do we go from prodromal synucleinopathy to prodromal MSA? can we in some way fine-tune the diagnosis? So how do we do that? Well, we added exclusion criteria in the diagnostic criteria of prodromal that are biomarkers suggesting that we know that good data that these biomarkers suggest phenoconversion to Parkinson's disease or dementia, some Lewy body disease. I'm going to focus on the first one, which is loss of sense of smell or anosmia. Also, an MIBG scan abnormal also suggests Lewy body diseases, but not everybody has these. And of course, fluctuating cognition, visual hallucinations, or dementia within 3 years is also an exclusion criteria. I'm gonna, none of these fluctuating cognition or visual hallucinations were in those patients were excluded before. So what we wanted to do was saying, okay, can we use this data from the natural history study to in some way estimate sensitivity and specificity of these diagnostic criteria possible prodromal MSA? So what did we do? We said, okay, let's reclassify the patients with this new diagnosis, which is possible prodromal MSA. We got all patients with pure autonomic failure, and we looked who had at what time subtle motor signs and no anosmia, those three things, and we classified them as possible prodromal MSA. Out of 118 patients with pure autonomic failure had complete data, we had more, but complete data, 118, 57 qualified, 61 did not. The ones that qualified had NOH, subtle motor signs, and no anosmia with an UPSID. UPSID is that scale, so in a second, that had to be higher than 18, meaning lower than 18 is total anosmia. It had to be higher. That was the criteria. 61 did not. Of those 57, 16%, meaning 9, phenoconverted to MSA within 5 years. And the ones that did not qualify for possible prodromal, none of them phenoconverted to MSA. So look here, you have the other phenoconversion. Right? Now, based on this, we said, okay, let's do a simple. Look, I wanted to do multivariate analysis. The statistician, I need a better statistician, said, you cannot do this. We argued for 2 months with these numbers. But we said, okay, we'll do simple statistics, which is at least the ones I understand. So let's do sensitivity and specificity. We had on these patients true positives, meaning had the criteria 9, false positive 48, false negative 0, true negative 61. That gives us the formula, true positive divided by true positive plus false negative, gave us 100% sensitivity for the diagnosis, but quite low specificity, meaning we captured all patients, but we also captured many that didn't develop the disease, so specificity was low. So we said, okay, can we, with low tech, can we improve this? So we thought, okay, now what you see at the right of the slide is the UPSID scale. You see that we were allowing in the definition a good amount of smell loss, meaning is no anosmia. The decrease in smell is not an exclusion. Patients have to be right above this blue line. Now, that captures all patients who will phenoconvert to MSA, but many who will not. So we said, what if we use more strict criteria? And let's take the UPSID, which is easy. What if we go up? We ask, okay, it has to be better than 18, but it can be let's say 28. So we took here moderate microsmia, right? And we changed the diagnostic criteria, still no anosmia, but more preserved smell. That gave us different numbers. 24 patients had prodromal MSA, meaning NOH, subtle motor signs, and an UPSID, meaning smell of equal or higher than 28. 94 did not qualify, meaning had NOH, but had no subtle motor signs or an UPSID below 28. If we do that, what we see is that 29%, 7, phenoconverted to MSA on the group we expected. In the other one, only 2% we lost that phenoconverted to MSA. Right? Now, if you look at these, what we were able to do, we increased the false negatives a little bit, just 2%, but we decreased the false positive quite dramatically from 48 to 17. So if you do again the sensitivity and specificity, I put in red the numbers for an upsit of equal or higher than 28, and in black, the classic 18. As you see, with this new way, we lose some, we decrease the specificity a bit, but not too much, to 84%, but we increase sensitivity quite dramatically to 78%. So we said, okay, we believe possible prodromal MSA with an upsit equal or higher than 28 loses sensitivity, as I mentioned, from 100% to 78%, because some patients with MSA have decreased sense of smell. They don't have anosmia, but they do have decreased sense of smell. And in fact, we just had 6 months ago a patient with decreased smell, no anosmia, but decreased smell, and had autopsy-confirmed MSA. So we increased specificity from 56% to 84% to 84%. I think perhaps is a good compromise. So to compare, meaning how different is this from the other categories? Remember, clinically established and clinically probable are two well-defined criteria, and they're not research criteria. This one, the one I'm talking about, is a research criteria that has not been validated, requires validation. So, but if you compare them, I use the same paper Vasilios was showing with the pathological validation of the MDS criteria using the brain bank. Do you notice that all the papers validate criteria for, well, the ones available for clinically probable and clinically established? Nobody even touches possible prodromal, perhaps because movement disorders clinic do not see possible prodromal. They see Jales, Paniker sees them, and we see them also because we do movement disorders and autonomic, right? So, but if you look at the numbers, at least with this very simple validation criteria that I showed you, the numbers are not that different. From this pathological validation, this is for early diagnosis, sensitivity and specificity, you see them here. For clinically probably 62%, 95%. For clinically established, very low sensitivity by high specificity. Now, if you look at the MDS prodromal, if you have with upsit equal or higher than 18, 100% sensitivity, low specificity. With the other, what I told you before, 78, 84%. You see, they are not that far. They are quite similar to the other better known criteria. So in conclusion, possible prodromal MSA is a useful diagnostic criteria that we believe will allow early diagnosis, and we would like to advertise it, that everybody is that, these, and I've been arguing this about a long time, and this is where we should treat patients. Ideally, clinical trials should not go to clinically established and clinically probable. They should go here. If we are able to build a straw man with high specificity and high sensitivity that this is MSA, this is the time to treat them, because the drugs we have do not revert the disease. The drugs we have stop progression, at least what we know now. Stopping progression before the motor symptoms occur makes sense. Stopping progression after, of course, makes sense, but it's not the same. I want to tell you that there are several other features that that also increase the odds, but the numbers are very small in the amount of data to make good predictions. And statisticians, although I can do it, refuse to accept analysis with such small amount of data. But plasma norepinephrine high increases the odds. Higher supine heart rate increases the odds. Bladder retention increases the odds. And younger age at onset, in all statistics, when it's in the 50s, increases the odds. Now, clearly, sensitivity will increase with the use of NFL and alpha-synuclein aggregation assays, probably in the future. And in fact, there's a patient from the Mayo Group, from Wolfgang, from a few years ago, showing on the left you have neurofibrillary light, neurofilament light, and on the right you have the fluorescence of the aggregation assay. And here they added MSA, confirmed MSA, controls PD and DLV, and pure autonomic failure. As you see, the ones in red are the ones with pure autonomic failure that then phenoconverted, only 5 patients, and had higher levels from the beginning. Same story with synuclein aggregation assay. You see that the ones with PAF that phenoconverted to multiple system atrophy here in red had this low pattern, this original low pattern of in the curve of fluorescence. So why is early diagnosis important? Well, you know why, right? I mean, everybody would be happy with early diagnosis because it opens a crucial time window for earlier intervention. And it gives patients the opportunity to enroll in clinical trials, either disease-modifying therapy or symptomatic therapy. I'm going to end with a few slides. Viorica and Valeria asked me to cover prodromal, but also one of the trials. How could I do that in a short time? I tried. I only have 2 slides. The disease-modifying therapies you heard about. I'm going to talk about symptomatic therapy just a little bit to remind you that there is a drug recruiting for clinical trials now that is personalized medicine directly designed and thought for multiple system atrophy. Now, as you know, in that early prodromal phase, the most disabling symptom at early stage is orthostatic hypotension, and many patients, despite available treatment, remain symptomatic. In fact, 85% of patients are still symptomatic despite treatment. So ampreloxetine, what I just want to give a pitch for a minute, is a norepinephrine transporter inhibitor specifically tailored for multiple system atrophy, which is currently in clinical trials and is recruiting only MSA patients. Valeria is leading the trial here in Queen Square. Send her the patients. I'm going to tell you why. You know, neurogenic orthostatic hypotension is due to reduced norepinephrine release from postganglionic sympathetic neurons. Those are the neurons that go directly to the blood vessel. Norepinephrine is not released appropriately there. Now, if you remember from school, even if you're not interested in autonomic, there are really 3 neurons that go from the rostral ventrolateral medulla, right, where the pacemaker of sympathetic neurons are. The first neurons goes to intermediate lateral cones in the core, then the preganglionic to the ganglia, and from the ganglia goes the other neuron directly to the blood vessel and releases norepinephrine. Interestingly, and quite surprisingly, in patients with multiple system atrophy, that last neuron, the postganglionic neuron, the one that releases norepinephrine, is quite spared, but it doesn't release norepinephrine. Why? Because the previous neurons are the ones severely affected. So the neurons that last neuron is sort of spared, but it just doesn't get the signals. That gives us a big opportunity to improve or enhance the effect of that last neuron. How do we do that? We can do it with the norepinephrine transporter inhibitor. Why? Well, I have this animation, which I didn't do, somebody did, but I think it's pretty good. It shows that you have a normal neuron, action potential, sympathetic nerves are activated, they release norepinephrine. Norepinephrine binds to alpha receptors in the vascular wall, produces vasoconstriction there, and then quickly there is reuptake, is used again, that norepinephrine comes back to the preganglionic, to the presynaptic, meaning the postganglionic neuron, it's taken back there and is reused. Now, in multiple system atrophy, that neuron is spared, but there are less action potential, less activation. So with the less activation, less norepinephrine release, but still norepinephrine release. Vasoconstriction, and what happens? If we inhibit the reuptake here, we increase the amount of norepinephrine in the synaptic cleft that increases vasoconstriction and enhances the effect of norepinephrine. this is not just a theory. In the previous trial to the one now, patients with all, now we're only recruiting MSA. In the previous trial, it was all synucleinopathies. And as you see here, the increase in norepinephrine was much more significant, as expected, in patients with MSA than in PD or PAF. And the DHPG, which is the intraneuronal metabolite of norepinephrine, decrease more or decrease significantly in MSA. Decrease in all of them, it's telling you that there is less intraneuronal metabolism. Not only that, the effect in blood pressure, I'm doing this very fast, was significant only in multiple system atrophy, blood pressure standing. So this gave us the impetus to say, this drug is tailored. I mean, this was the original idea. We wanted to compare that the drug is specifically tailored for multiple system atrophy. In all symptoms also, and this is a forest plot showing the difference. This is just for the MSA subgroup on the initial trial. And as you see, everything that is in blue favors the drug, and most of the symptoms of this orthostatic hypotension questionnaire that we used to rate the symptoms, all of them favor, most of them favor ampreloxetine. So there is strong evidence. What we need now is to prove that it works in a large phase 3 clinical trial and send the patients to Valeria. Please, we have to, this is the only one, the only drug specifically tailored only for MSA, for treatment of MSA. Thank you very much.
So much, Horatio. And now we move on the second talk, and then we will have a session for few questions. So I will introduce Professor George Panica, head of the Neuroneurology unit here at Queen's Square. Professor Panica has done an immense amount of work in neuroneurology, and it has a very strong interest in patients with multiple system atrophy. And he will discuss today blood dysfunction in patients with MSA, which is a very prominent feature, but also what the imaging of the spinal cord can tell us regarding the pathology and how can potentially actually inform clinical practice.
Thank you very much. Thank you, Valeria, for this. Thank you. And thank you for those warm words of introduction. So my name is Dilesh Panik. I'm a neurologist with interest in neurogenic bladder, bowel, sexual dysfunction. These are my disclosures, and in the next 20 minutes, I'll be sort of taking you through a journey in terms of urinary problems in MSA and how imaging may help understand this further. We've been hearing this morning from Vasilis and from Horatio about urinary problems and in general urinary symptoms. And what we've understood is that urinary problems are quite common. In fact, the commonest presenting symptom in multiple system atrophy. And when we say urinary problems, this can span on one end problems with storage, resulting in urinary incontinence, and problems with voiding with urinary retention. And because of the overlapping nature of urological and gynecological problems, often the bladder problems, or the neurogenic bladder problems in MSA are mistaken for other sort of conditions. And in fact, more than 40% of patients with MSA presenting with urinary symptoms are misdiagnosed with a urological problem such as prostate enlargement, often undergoing different sort of treatments, including surgery, predictably with very poor outcomes. Now, we know that urogenital autonomic dysfunction and cardiovascular autonomic dysfunction are about a spectrum. But what we find is that though it is sort of autonomic dysfunction resulting in failure, that actually there are differences in terms of the onset of urogenital and cardiovascular failure. And when in studies like these, where patients are asked about urinary symptoms and orthotype sort of symptoms, what we often find is that urinary symptoms and genital symptoms may actually predate the onset of symptoms from the cardiovascular symptoms. Now, the diagram on your left is essentially really a schematic of the neural control of bladder functions and health. So you have sort of different circuits, forebrain circuits, sort of which feed to different brainstem nuclei in the midbrain and the medulla, and also you have sort of neural pathways between the brainstem and the spinal cord, thereby ultimately resulting in the control of bladder functions. Now, if we study the pathological changes in MSA, what you find is many of these changes will overlap with these same regions that are involved in the neural control of the bladder, particularly regions in the neocortex, your basal ganglion network, different parts of it, the cerebellum, at the level of the brainstem, in midbrain, particularly the periaqueductal gray. The pontine micturition center is in the pontine is one of the pontine tegmental nuclei, which are particularly susceptible in MSA. And what is unique about MSA compared to other sort of neurodegenerative conditions is the involvement of the spinal cord. And we heard about sort of the preganglionic efferent sympathetic and parasympathetic output at the thoracolumbar cord and the parasympathetic output of the sacral spinal cord. And there's a predilection for degeneration of these neurons, but also in the anterior horn cells, particularly in the sacral spinal cord, where you have sort of hybrid anterior horn cells, which are sort of have sort of characteristics of autonomic neurons, which we call the ONIF nucleus, which is particularly susceptible. The ONIF nucleus innervates your sphincter muscles, the urethral and the anal sphincter muscles. So reflecting this complex pathophysiology is also the complex presentation of bladder problems. And so some patients can have a top-bottom presentation where they present with predominantly storage symptoms early in the course of the disease. They have a rather efficient voiding, and therefore usually the post-void residual volumes are low, and they have what we call detrusor overactivity neurodynamics. Now, patients, as they start developing spinal cord problems, they start to develop storage and voiding problems. This could also occur from brainstem dysfunction as well. And patients have either underactive bladder, what we call an underactive detrusor, or detrusor sphincter dyssynergia. Now, there are some patients that we see who we think may have a bottom-top presentation, where they present predominantly with the sacral cord pathology, and they present predominantly with urinary retention, and post-void residual volumes are often elevated, and urodynamics will demonstrate a contractile detrusor. And I'll illustrate a case just to sort of share our experience with this sort of cohort of patients. So, and there are different treatment options available for managing the urinary and sexual problems in MSA. This is just guidelines published just 1 week ago. Actually, this is I would say it's a massive piece of work with the European Academy of Neurology and the International Neurological Society and the European Federation of Autonomic Societies, where actually we have developed guidelines for the very first time for neurologists, so that's the non-urologists, of course, in assessing and managing of urinary and sexual symptoms in neurological disease, which includes conditions such as MSA. Also reflected in this sort of lower urinary tract dysfunction, the complexities that in the diagnostic criteria of MSA, we have both ends of the urinary problem. So you have sort of unexplained voiding difficulties with a high post-void residual volume, and unexplained urinary urge incontinence as grouped as sort of core clinical autonomic features. Now, these symptoms are quite wide, but actually differentiating sort of early MSA from Parkinson's can be a challenge. And when we look at sort of the analysis in the lens of the urinary problems, the commonest symptom that patients will tell you about or will volunteer, and the one that's actually it's more easy to ask in a history in the clinic, is about incontinence. But you find that storage problems, urinary urge incontinence, has actually dismal sensitivity and specificity to distinguish from MSA and Parkinson's disease. On the other hand, urinary retention is much more specific. And though the sensitivity is low, and thank you for Horatio for explaining the differences between the two, but you can find that the specificity is rather high, which means that it's very good in terms of identifying sort of the true negatives if there is no urinary retention. We know that in the journey of the patient, urinary retention gradually progresses. It's a relentless condition, and eventually regions of the brainstem of the spinal cord that are involved in the voiding reflex are involved, and patients will develop voiding dysfunction. What we find in sort of in cross-sectional studies is that roughly patients who are roughly around year 2 of their disease in terms of the neurological symptoms, more than 50% of them will be having a level of urinary retention that crosses a threshold for commencing intermittent self-catheterization. And you can find that as the number of years of onset of illness progresses, that their urinary retention increases. So early spinal cord involvement in MSA is really something quite interesting, because these patients will present with predominantly autonomic problems, cardiovascular autonomic failure and urogenital autonomic failure, and urinary retention would tend to occur quite early on in this group of patients. Now, there is evidence from autopsy studies from way back in the 80s that have demonstrated significant pathological involvement in the sacral spinal cord. And this is work in sort of when it was called SDS, showing a depletion of neurons and gliosis in relation to the intermediate lateral horn and in relation to the anterior horn, which would be sort of in the area of sort of the nucleus. In transgenic mouse models, which have been extensively investigated by Nadia Stefanova and her team in Innsbruck. Once again, there was very early involvement of the intermediate lateral horn of the spinal cord, as well as the ONF nucleus. And in this study, where of course longitudinal sacrificing was possible, that actually changes in ONF nucleus were seen as early as 2 months of age in these sort of in these transgenic mouse models, indicating that this region can be involved quite early on. And this is just in clinic how you might identify or might actually start thinking about a patient who can present maybe with the sacral cord first presentation. And this is sort of a patient who was referred just because from a urology team actually saying there's unexplained urinary and sexual problems, as you can read here. Is there anything neurological? And so as a clinician, what are the tools at your hand if you want to investigate the sacral spinal cord? So you really would like to study the sacral spinal cord, the intermediate lateral horn, so your bladder motor neurons that innervate the bladder, but also the ONF nucleus, which innervates the sphincter. And so the patient underwent pelvic nerve physiology studies, and the anal sphincter EMG was significantly abnormal. And the patient underwent the urodynamic study, which showed there was an atonic detrusor. So we knew at this point that the patient was having a neurological cause for their retention, absolutely no neurological symptoms and signs, no history of RBD, MRI brain was normal. We didn't do a DAT scan at that stage. And of course, we didn't involve malaria at that point because the patient's blood pressure had been normal, at least in clinic. But we did sort of agree that there was something neurological. We did suspect it may eventually turn out to be something neurodegenerative, and the patient was followed up on an annual basis. And what we found was within 7 years of onset of their symptoms is that they began to develop their very first neurological symptoms and signs. And after that, it was actually a rather quick progression, unfortunately, and the patient passed away. But that was sort of 11 years into the onset of their urinary problems. And the autopsy actually showed pathology that was consistent with multiple system atrophy. So this is all good because, you know, we have some tools, but we need to think about in vivo, is it possible to assess the lumbosacral spinal cord? And so there are functional tools and there are structural tools. So functionally, we can assess this through pelvic nerve physiology tests. We can innervate the innervation to from the ONF nucleus, or urodynamics, which gives you an indirect assessment of bladder functions. Or what about a structural assessment and whether a high resolution MR imaging is possible to image the lumbosacral spinal cord. So first about pelvic nerve physiology testing. So it's not just about EMGs, but essentially any innervation from the ONF nucleus to the pelvic floor could be assessed. So it could be anal sphincter EMG, or it could be EMG the urethral sphincter, or the EMG the bulbar cavernosus, and we sort of recognize the motor unit action potentials, and based on the morphology, we can then say if it's normal or abnormal, or we can assess what's called the bulbar cavernosus reflex between the sacral spinal cord and the pelvic floor. Bulbar cavernosus muscle particularly, and we record a response and measure the latency. And there are numerous studies that have evaluated these tests in the context of sort of MSA versus Parkinson's in terms of making that distinction. And you find that sort of it's modest sensitivity and specificity for anal sphincter EMG. But there have been studies that have correlated the changes in EMG of the anal sphincter with urinary incontinence, but also with urinary retention. And with the bulbar cavernosus reflex, slightly better in terms of specificity, but once again, low sensitivity. Now, this is where we were looking at other tools, and Mario Sinakis is in the audience here, and his team had actually been interested at their NMR unit at Queen's Court. We're actually developing a high-resolution structural imaging for the lumbosacral cord. And in their work, they sort of overcame the challenges involved in imaging this region, essentially because of challenges in immobilization of the patient and also controlling for the different physiological variations that can occur and the physiological motion, and developed this protocol, which using sort of a reference point that can be readily identified in MRI, that is a lumbosacral enlargement. And by identifying the lumbosacral enlargement, they were then able to validate a technique to segment the gray matter and the white matter. And of course, then it became, what is the clinical utility? So that's when we began to collaborate with the team, and we said, well, look, this is a lumbosacral enlargement, but in conditions of MSA, we're actually interested in something slightly more caudally, which is associated in the conus medullaris. And so the next piece of work was actually applying this and then extending this to the to much more challenging region, the conus. Challenging mainly because of the variability in the length, but all of that region, but also in terms of the sort of the tapering nature of the conus. But this was actually a successful collaboration. In fact, from this work, a validated technique has now been established for actually imaging. And you can see that the gray and the white matter can be reliably sort of segmented in the conus medullaris. So this then led to the question, what happens in multiple system atrophy? And this was sort of a collaboration between uroneurology and Marius' unit and with Henry Holden. And so patients who had sort of probable MSA, who had an abnormal anal sphincter EMG were chosen. So these are patients that we know clinically have some ONF nucleus involvement. So it was really a proof of concept. And what was demonstrated, so these are essentially 0 is a lumbosacral enlargement, and anything minus is sort of towards the conus medullaris, as you can see in the schematic here. And what was seen is that there are, you know, a predictable sort of reduction in the gray matter and white matter fraction going caudally, but there was a significant sort of reduction in the gray matter volume fraction in the LSC, but also the conus medullaris in patients with MSA compared to sort of age-matched healthy subjects. And what was also seen, and when we applied different urinary symptom questionnaires, is that actually the voiding symptoms, the voiding difficulties the patient had, significantly correlated with changes in the gray matter cross-sectional area, both at the level of the lumbosacral enlargement and the conus medullaris. So then this actually leads to to more unknown. So actually, when do these changes actually begin? And do they occur in all synucleinopathies, or is it specifically the group of patients, say, with PAF that actually will phenoconvert to MSA? And can identifying sacrospinal cord changes early in the disease help in predicting phenoconversion to MSA? And we heard from Horatio about sort of the journey of PAF, where it's often just a transit point for a third of the patients who then could sort of phenoconvert to either more likely PD-DLB, but around a quarter of the patients MSA. And so this has sort of led to our next study, and delighted that we're also collaborating with Valeria and the autonomics team. And we are very grateful to the MSA Trust for funding this sort of exploratory study. Now, you might be wondering what happens in pure autonomic failure. And I suppose on the back of the collaboration with autonomics, we've been able to establish that patients with pure autonomic failure, they do have ONF nucleus changes. This is just the example of a patient where you might recall the bulbar cavernosus reflex you saw on the earlier slide. Here it's actually an absent response. And the anal sphincter EMG was abnormal in this patient who had a clinical diagnosis of pure autonomic failure. What we've also demonstrated is that the neurophysiology changes deteriorate over time. So you do have a biomarker that you can accurately follow up in a very minimally invasive manner. And so this 61-year-old lady who presented in 2021 with PAF, typical symptoms of PAF, and met the criteria, their anal sphincter EMG was normal. However, their bulbar cavernosus reflex was present, but the latency, which we can accurately measure, was actually prolonged and was abnormal. The patient returned 2 years ago, and that normal anal sphincter EMG became abnormal, and we could no longer record the response on the right side. And the bulbar cavernosus reflex that had just had an abnormal latency had now disappeared completely. So we knew clinically that actually these patients do progress in terms of the ONIF nucleus degeneration. So in this study, what we propose is that patients with pure autonomic failure who are with Valerius QSA prodromal study and had a greater risk for phenoconverting to MSA or DOB will be included. They will undergo clinical assessments plus high-resolution MR volumetric and quantitative imaging of the lumbosacral enlargement and the conus imaging. Plus, they'll be undergoing pelvic neurophysiology. And these patients will be annually followed up in terms of their neurological scores, cognitive scores. They'll be also having autonomic and pelvic visceral questionnaires addressing bowel, bladder, and sexual symptoms. They'll be having their post-forward residual volumes measured and also autonomic function tests. And in this collaborative study, the patients will be having a total follow-up of 6 months, of 6 years. And what we aim to look for here is to evaluate changes in the cord structure in patients presenting with a clinical diagnosis of pure autonomic failure using MRI-derived measures, but also to assess whether the changes in the spinal cord can actually predict those who phenoconvert to MSA, and to correlate the changes that we see in the lumbosacral cord with pelvic neurophysiology findings, and also the bowel, bladder, sexual symptoms, and autonomic dysfunction. So in conclusion, urinary problems occur early in MSA. The complexity of urinary dysfunction that you've heard today actually just reflects the very wide distribution of pathology in the CNS that occurs in MSA, affecting different regions that are involved in the neural control of the lower urinary tract and help. That the sacral spinal cord is particularly susceptible to involvement, which makes this condition unique compared to other neurodegenerative conditions. That urinary retention, we believe, can be initial presentation in the cohort of, and a subset of patients, and that one can actually image or assess the lumbosacral spinal cord functionally by pelvic neurophysiology and urodynamics, and structurally through high-resolution MRI imaging. And preliminary evidence does suggest that these tools, that from these tools, that actually abnormalities can occur early in the stage. Thank you very much.
Thank you so much, Giles.
I think we have time for 1 or 2 questions, and then also remember our speakers will be available during the refreshment break, so you can always grab them then.
So 1, 2 question.
That lady which you showed, the one with very minimal features, a DAT scan at that stage would have, I'm sure, shown that she had an abnormality. Now, whether she could turn into Parkinson's or MSA is debatable, but that may be an additional tool. And I think Valeria has done some studies on this, which is important. The other thing I think is you mentioned the synuclein assays, which will obviously help us. And the Japanese are suggesting that they can differentiate between the synuclein of MSA versus synuclein of PD. And I don't know whether you're carrying on that work, or perhaps even Andrew Singleton might tell us whether this sort of assays are being considered. So I think a DAT scan and particular synuclein.
So the answer, Kailash, is yes to both. We didn't have that scan on that lady. The idea was to, well, I think that scan would be very useful, and synuclein aggregation assay would also be very useful. I'm sort of partial to clinical features and low tech. You know, a smell test is $25. A DAT scan in America is $800. So this you can do in any place with just with your clinical acumen and the smell. And I do agree with you that that scan could be very useful. And perhaps MRI, we don't have enough MRIs early on. That's another field that we are developing, and the same with the synuclein aggregation assay, the same with NFL, that seems to be higher, and perhaps even proteomics, what Giles just mentioned, maybe at the beginning, because this woman had urinary retention 2 years before motor symptoms. So as Giles says, something was wrong there. The on-off nucleus had to be involved, and her blood pressure dropped dramatically. the rostroventrolateral medulla and the intermediate lateral nose had to be involved. There's no other. So the answer is yes.
Any other question?
The application to me on an application with MSA. And he was passing you very well, but lately we developed tension. So much that we have to capitalize itself. But then it's so that on 250, 300 mils, he can't pass. But when it comes to 400, 500, he doesn't. Got mental freezing and also some mobility freezing. Could the bladder be also acting as frozen reaction rather than purely autonomic problem?
I'd be happy to try to answer, but we have an expert here. So that is, you can.
Well, there isn't an easy answer to that because what happens is that contrast to Parkinson's, where you have a bradykinesia of the pelvic floor, with MSA you have a denervated pelvic floor, and you find that actually the entire pelvic floor becomes loose. I suspect what's happening in your case is that you're building a pressure head. And so from 100 to 250 to 400, the pressure is rising in the bladder, and you have a weak outlet. And so the pressure is going to overcome the outlet, and so the patient will void. But this is actually a very poor voiding, and the patient is actually probably pressing in, bearing down, or just by gravity getting drops of urine. So it isn't a sustainable way of management, but I suspect that's what's happening. And many times you find that patients with MSA are unhappy when they go to the urologist because they're managing their bladder with their retention, they're peeing, but when someone goes and puts a camera in or the catheter in, that pressure head is no longer there and they go into complete retention. They blame the urologist, but actually it's just that pressure head has been removed.
Didn't we call that for the non-urology similar to overflowing continence?
Absolutely. So it is
overflowing continence.
it's probably overflowing continence, but was perceived as voiding.
One of the things which we should be doing is educating urologists, not neuroradiologists. So we want to look at, go around and tell urologists, the guys who are the surgical guys, you know, who normally sort of don't listen to anything medical, to explain to them that there is this condition. I mean, if you're trying to look for prodromal patients, that's the area we got to tap. We got to go and educate the simple urology, if I can use that word, simple in them.
Kailash, there's an example, which is orthopedic surgeons with amyloidosis. You know, when we found out, well, I mean, a lot of people, not me, that amyloidosis can produce carpal tunnel and can be a presentation of carpal tunnel.
Mhm.
Now the orthopedic surgeons that do carpal tunnel surgery look for amyloidosis. And it's incredible the amount they found, and they didn't even know what amyloidosis was, which is what you were saying with regular urologists. Tell them, you guys are seeing early MSA, and there may be other, right? There, achalasia, there may be many other prodromal syndromes, phenotypes that we have not recognized that are due to synuclein accumulation, and we are not seeing them. We see autonomic because they come to me, or they go to Jalish for urology. I bet you there are at least 5 other phenotypes with that doctors that they see that they don't connect with that.
And you got to educate.
Correct.
Very quick reply. So there's a big interest in occult neurology in the urology world now, and so whichever urology meeting you go to, there will be a session on occult neurology and what the urologist misses. The second general piece of advice is to actually reach out to the right type of urologist. They're all in different sort of breeds, but it's a functional urologist. Well, it's a functional urologist. You don't want to be going to a prostate specialist or to a stones or a cancer specialist. It's a functional urologist or a female urologist. These are the ones who are probably going to see your very early MSA patients. So Benjamin Butler, for example, NYU.
Are much better. That's what you're saying.
So female urology by male and female doctors, yes.
Okay.
And I suppose it's a similar challenge than obviously in the neurogenic age we had 10 years ago, no? Then patients they were referred to cardiologists, but they obviously were not identified as a neurogenic in nature. So it's a similar challenge. I guess the very last comment, just 2 seconds. I guess we've heard a similar narrative when it comes to different biomarkers. Yeah, so on the bladder side, the smell, the neurogenic OH, catecholamines. I guess the challenge is I don't know what you think about, but I don't think we have reached the state where we can say there's one biomarker that define this pathology is multifocal. So I guess we need to start thinking about a cluster of biomarker that will increase sensitivity and specificity. Otherwise, we're going to replicate a similar narrative. So these patients are it's a complex pathology. So maybe one story doesn't fit all of them.
Amyloidosis. In terms of the variability of clinical presentation, according to the site of accumulation of the protein, is another proteinopathy, right? I mean, it's the same story. It just doesn't go to the brain, the peripheral, particularly TTR. But it's misfolded protein that deposits. Of course, intracellular versus extracellular. But the idea is very similar.
And I think we just need to appreciate that every MSA patient is different, and there's so much of heterogeneity, and it's sort of like the proverbial elephant, you know. Yeah, you'll be seeing them for the blood pressure, you'll be seeing them for the bladder, someone else will see them for the smell maybe, but it is very much, you know, and so that's why I agree, there's no way that we could have one single biomarker because the pathology will start in some part of the neuroaxis.
Thank you. Thank you.
Hello, everybody. We're going to start our second session or our after tea session, third session. Just to say Verika's on the program twice, but please ignore that because she's extra special. So we thought we'd flatter her. But so Verika's up next. I've known Verika many years, and she's really one of the driving forces of MSA research in this country and internationally. She's got a lot of energy. She wants to talk to you all. She wants to work with you all. And of course, that's what we want as well. She's very collaborative and she's done some excellent work. So she's up first to talk to us about biomarkers. Good.
A lot, and today I have the task to provide an overview of the current state of research on fluid biomarkers, but also what this really means for patients' care today and for the future. We've heard already this is an aggressive, degenerative, fatal disease, really, with two main clinical phenotypes, Parkinsonian, cerebellar, but frequently overlaps. The two of them coexist in the same patient. But it has a rapid progression, and people die within 9 years in average. And what's worrying is that the usual average diagnostic delay is about 3 years, and this is time we simply don't have in this like fast-moving condition. There is no definitive diagnosis during life, and this is where the biomarkers come in. It's currently during life, the diagnosis relies entirely on a clinical judgment with some support from imaging. But imagine having a blood sample or a CSF type of test that would help us diagnose the patients earlier or predict prognosis or monitor disease and stratify those patients in clinical trials. And this is the promise of biomarker, and this is what the focus of this talk will be. We'll start with what has been done so far. Fluid biomarkers in MSA, they've been kind of focusing on the main areas of pathology, so biomarkers of oligodendroglial dysfunction, of neuronal loss, or alpha-synuclein, or inflammation. But in a way, I would suggest you ignore this slide because most of this work was either inconclusive or was showed inconsistent results. So it is, we don't have any really quantifiable biomarkers for alpha-synuclein, for either total alpha-synuclein or phosphorylated alpha-synuclein or the alpha-synuclein ratios between various species. So the only biomarker that has been validated consistently across neurodegenerative conditions, including MSA, was neurofilament and light chain NFL, and promising results are now coming from more qualitative type of assays, the seed amplification assays. The NFL, you've heard a little bit before from both Vasilios and Horacio. We've gathered this international collaboration with loads of samples, and we showed that there is a significant difference between healthy controls, but more importantly, it can track disease progression, both correlating with validated rating scales such as UMSAR, but also with disease milestones, and therefore kind of providing the evidence of its utility for drug trial, for monitoring response to treatment, hopefully. And also others and us have shown that there are differences between MSA patients and Parkinson's disease patients, but also with other conditions like spinocerebellar ataxia. So it does show a correlation with the level of neurodegeneration in MSA. It also correlates with the atrophy in the brain, and also with those regions that are of interest to MSA, such as the pons, striatum, middle cerebellar peduncles, but also with the rate of atrophy over time. So it's a good marker for kind of measuring how fast the disease changes over time. But it's not specific to MSA itself. Again, it correlates with, it's associated with survival, so higher NFL, shorter survival. We've looked at the trajectories, showing that the NFL initially rises as the disease gets worse, but very late in the disease, it starts to decelerate, but it still remains significantly higher than in healthy controls, and that it can help reduce the number of patients needed to show changes for clinical trials. Now, when taking this forward, we're looking in our own cohort, as Vasilios has presented, the French cohort. We have a similar setup here with Prospect M study. So we're looking at how can we inform prognostication for mostly not on an individual basis, not necessarily to tell the patient how much you've got when we see them, but for perhaps helping with advanced care planning and with a stratification for drug trials. So we looked at factors, negative factors of survival, and my colleague here, Dr. Go, has spent a lot of time and effort and thought onto this. But here I'll just present the two time points that are relevant for clinical practice and for trial. And this is survival from disease onset and survival from baseline clinic. So from disease onset, amongst the most important one that came out, and glad that Vasilios has shared very similar findings in his data. This is all unpublished data from both cohorts. So we're finding similar things, such as severe dysautonomia in the first years from onset, falls and walking aid use in the very beginning are individual predictors for like a negative survival outcome, but also the association of the higher NFL, so NFL above median, and NFL, and another marker we're looking at, the neutrophil lymphocyte ratios in the first 3 years. Now, for clinical kind of use, we're looking at survival from baseline visits. The ones that I think are worth knowing about will be a high UMSAR score at the baseline visit, is a significant predictor of negative survival, as well as a high NFL visit at NFL at that baseline visit. So currently we're working on putting this information together, so the clinical and the biomarker data into a multimodal prediction for negative survival outcomes in MSA. Now I move to alpha-synuclein, which is the protein of interest for MSA pathology. I mentioned about the challenges about quantifying the alpha-synuclein, but this is where this assays a more qualitative kind of approach come in. The seed amplification assays, they detect the misfolded alpha-synuclein by amplifying the seeds that come from the patient using the recombinant alpha-syn. And then this aggregation is tracked with a thioflavin fluorescence. And it's just kind of to mention that the field is advancing really quickly. There are new variations of the seed amplification assays, and I won't go into details. It's just to say that they have different practical applications, and we may have more tools to work with, hopefully, in the not that distant future. So what we know about the usefulness of the assay for MSA, there is enough robust data to show that actually the seeding kinetics, so the pattern of this amplification, is different in PD and MSA. And this in the first part of the slide, this is using CSF from patients with PD and MSA. It shows that the patients with MSA, they reach this plateau a lot lower, so they have a lower plateau compared to Parkinson's disease patients, which kind of suggests there is a more aggressive kinetic pattern in MSA, and that the MSA strains, the PD strains are different. And this indeed has kind of been supported by cryo-EM experiments, which shows that Parkinson's disease filaments have different protofilaments. They fold with like 8 beta sheets, and the MSA ones fold with 7 beta sheets. And within the MSA fibrils, there are 2 types, type 1 and type 2, and they can coexist with different in different ratios in the same patients. But in terms of diagnostic performance, so that what does it mean in clinical practice? This study has a large cohort of CSF from MSA patients, including path-confirmed, have cohorts of MSA patients recruited part of research studies where there is a lot of clinical data follow-up, and MSA patients recruited as part of like just a normal real life MSA clinic. And it shows that the assay is very good for distinguishing PD from healthy controls. Not as good in the research setting, but still kind of acceptable and encouraging. But I want to just mention, when compared the MSA versus healthy control from the cohort, which is like the real life clinical cohort, the specificity there has dropped quite a lot. So while this assay is encouraging, really is encouraging for MSA, still needs validation for clinical practice. I want to mention, Horacio has mentioned about the SAA in autonomic failure, but similar results are we're getting from patients with REM sleep behavior disorder, where the SAA is performing quite well, which suggests that we can actually pick up this abnormal aggregations patterns in patients who then have MSA much earlier before the motor symptoms. So this is very encouraging, but needs validation before we can apply it in clinic. And I won't talk about it, just want to show that all the patterns that you're used to see, PD higher, MSA lower, that refers to CSF. In other tissues, the kinetics are different, which reflects different kind of activities of alpha-synuclein strains in different types of tissue. So there are challenges, the methods, challenges with the actual sample matrix itself, challenges with the protocols. So I think as a community need to work together to overcome this and get the assay to perform at least as good as it does for Parkinson's disease. And I welcome anybody who has any ideas and wants to work on this. If we can contribute to your work, we're happy to. If you want to join us, we're happy to involve everyone. Then just briefly, kind of emerging biomarkers for MSA. Now, the extracellular vesicles, DEVs, they are small vesicles that deliver this bioactive whatever signatures from the cells they come from, and you can actually use those to find out what they have inside their cargo. So looking at EVs from MSA and Parkinson's disease, when you look at vesicles that come from neurons and oligodendroglia, they actually have different cargo in them. So the amount of alpha-synuclein in the oligodendroglial coming from Parkinson's disease and MSA and healthy controls is different. And in our own data, we also find a different amount of alpha-synucleins from oligodendroglial EVs compared to healthy controls, and also significant difference from neuronal EVs. Then what we did, we actually combined this with NFL, and we show that while the EVs alone have kind of low diagnostic accuracies, the specificities and sensitivities improve when combined together. When we put all the ones that we found significantly different together with NFL, in the last slide, we get a really good differentiations between healthy controls and MSA patients. But we need to follow up this with other disease groups, such as Parkinson's disease. Going backwards. Now, yeah, this is just like an idea what I think we'll be doing in the near future. Well, we'll suspect a synucleinopathy, and then we'll have some sort of CSF, CD amplification assay. I hope this will reach that level where we can use it, and then perhaps look at the filaments themselves with other types of technologies to look at the structural analysis. Now, this is something we don't have, and something I would love to have. And Andy and his team has published this amazing paper on proteomic profile of Parkinson's disease, where they use proteomics, they combined it with genetics, they have stratified patients PD patients based on their profiles, and using the GWAS they found, the hits that then translate to the right proteins for Parkinson's pathology. So I would love to have something like this, and I think this is where our field should also move. We've done at a much smaller scale in MSA, atypical Parkinsonism. We've done proteomics analysis using Olink technology, and we've found differences between the atypical groups and the controls. At a subgroup level, we found differences between MSA and PSP and CBD, and we show that the performance improves when we combine the proteomics that the new proteomic signature that we find from this with NFL, which is consistently showing to be performing well. So by adding NFL to this new emerging markers seems to improve their performance. So yeah, we have barriers that we need to overcome. The sample cohorts, the samples, the lack of the standardization of assays, and looking for longitudinal multicenter studies. This is just a brief overview what we have here at UCL. We collect biomarkers, we have natural history study, we run clinical trials, we try to engage patients with genetics, imaging, and we look after our patients in our specialist clinic. And I'm grateful to the funding and the support we've received throughout the years to make this work possible. And this is the sort of data we have at the moment, and kind of the new papers that data is generating. And we encourage collaboration so that we can make use as much as possible of this valuable data sets and samples. So in summary, looking ahead, there won't be one biomarker, that's for sure. What we have at the moment is a combination of NFL, of alpha-synuclein, perhaps something similar with, or a variation of the seed amplification assay, and things like inflammatory protein, proteomics. We need to combine that with clinical and imaging markers to stratify our patients better and to look at how these perform into earlier stages, like the prodromal MSA stages. And I thank everybody who has supported us throughout the years, and our patients, our collaborators, and our funders. Thank you very much.
As always, we've got questions afterwards, but we're running a bit tight on time. Next, we have Zane. Zane is a neuropathologist, and as you all know, good neuropathologists are like gold dust. And Zane is one of those. She's going to talk to us about dual pathology.
Yeah, good afternoon, everyone. And first of all, thank you so much for inviting to present the work we've been carrying out at Queen's Square Brain Bank on MSA pathology and co-pathology, and very much taking on from where Viorica left. Over the years, we really, from the pathology point of view, have really learned and understood how MSA pathology forms from a native alpha-synuclein protein going through the monomeric, oligomeric, fibrillar stage, resulting in these inclusions we can see on the histology, which then subsequently results in brain atrophy. And as Viorica said, from the clinical diagnostic point of view, there are no definitive biomarkers one can use to diagnose these diseases. Well, there are some, obviously, you guys as clinicians can are using, but with no sort of major success, in particular in the early stages of the disease, such as MRI scan, which would really reflect that structural brain atrophy evidence we see at the postmortem, PET scan, for example, for alpha-synuclein, which would reflect from the pathology point of view these inclusions in different cell types we see on postmortem, or as Viorica explained in detail, the seed amplification assay, which from the sort of pathology point of view would very much reflect these protofibrillar, fibrillar stages. But because there are no definitive clinical biomarkers, of course, we all know there's so much more work we need to do. And we also know that it's not only the inclusion pathology where it stops, that there is a range of post-translational modifications associated with the native and misfolded alpha-synuclein, their gene expression signatures, there potentially are also co-pathologies, which all influence and therefore make it troublesome to either develop or interpret these biomarkers. And that's exactly from sort of pathology point of view, which we want to also help and see how we can address in a pathology setting, and employing digital pathology, specifically machine learning-based algorithms to quantify various different either misfolded protein, inflammatory or vascular pathologies, using spatial transcriptomics to really look at these molecular signatures. Nanopore sensing is something we are developing to specifically look whether post-translational modifications could be used as a biomarker in addition to all the other tests currently being in development. Using cryo-EM to look at the filament structure, whether that can help us to really understand the MSA disease better, which is in collaboration with the team in Cambridge. Using also cell seeding assays to really understand whether we can take further the understanding about the pathomechanisms, and specifically the thing we are most interested in is the phenotypic diversity and cellular and regional vulnerability, and equally using proximity ligation assay, looking at these very early oligomeric stages of the misfolded protein alpha-synuclein. And as I mentioned, it's really that those pathomechanisms which we need to understand better to actually develop biomarkers. And what we are particularly interested in is what are the reasons behind the phenotypic diversity and the cellular and regional vulnerability we see in across the MSA cases. And I'll start off with the cell seeding and cryo-EM evidence we have so far addressing specifically the issue around the phenotypic diversity. So as you clinicians all know, there are obviously this range of clinical phenotypes in a patient with MSA pathology. And from the pathology point of view, these clinical phenotypes, they really reflect the regional brain atrophy that in the MSA-C clinical phenotype, from the pathology point of view, there will be very obvious pontine base atrophy, cerebellar white matter atrophy. In the MSA-P clinical phenotype, the key macroscopic finding will be the obvious putaminal atrophy, and in sort of the rare sort of pathological subtype where we only see substantia nigra and locus ceruleus atrophy and no other these OPCA or SND region changes. We call it MSA minimal change, which macroscopically looks most like Parkinson's disease, but clinically can have various different ranges of phenotypes. And equally, there is also the pathological subtype of MSA FTLD, which really looks like frontotemporal lobar degeneration, where the key macroscopic finding is this really obvious medial temporal lobe atrophy, whilst the actual pathology is MSA. And again, clinically, these patients typically would present with frontotemporal dementia or cortical basal syndrome. So from these pathology studies over the years, we know that the clinical phenotype definitely correlates with the regional atrophy pattern. But what is the reason behind this regional atrophy variability between different patients, which then leads to different clinical phenotypes, is really the question we really want to understand. And with the cell seeding experiments and cryo-EM, what we have shown so far, that the phenotypic diversity is not explained by filament structure or seeding pattern. So here as an example is a case with a classic pathological diagnosis of MSA FTLD, very heavy medial temporal lobe atrophy, very heavy misfolded alpha-synuclein neuronal and glial pathology, specifically in medial temporal lobe regions. However, using the biosensor alpha-synuclein seeding line, what we can see here that this case, the FTLD synuclein case, it seeds identical to a classic MSA case rather than G51D mutant Parkinson's disease case or dementia with Lewy body cases. So that the seeding activity is no way different from any of the MSA classical SND or OPCA phenotypes. And the same at the cryo-EM level, that it turns out that the filament structure is actually identical in to MSA typical case in this MSA FTLD case, which really has not solved the question we had. What is the reason behind the phenotypic diversity? Yes, we know it's the regional involvement of the pathology, to some extent also the extent of the alpha-synuclein pathology in those regions, but it's not the filament structure. So really more work needs to be done to really understand what's the cause of the cellular and regional vulnerability, which then leads to the phenotypic diversity. And for that, we've used spatial transcriptomics to really try to see whether we can understand more about these reasons. And so there is also an interesting thing, which all pathologists in the audience would know, that in multiple system atrophy, there is obviously a clear difference between Parkinson's disease in terms of the cellular vulnerability, that in typical multiple system atrophy case, and here as an example, I'm showing the inferior olivary nucleus, but there are many other gray matter regions and white matter regions, of course, in MSA, which show very distinct vulnerability to alpha-synuclein pathology than it's in Parkinson's disease. And in inferior olivary nucleus, in every given MSA case, there will be way more neuronal alpha-synuclein inclusions compared to Parkinson's disease. There will be way more frequent threads compared to Parkinson's disease. There will be way more frequent glial cytoplasmic inclusions compared to Parkinson's disease, which clearly is evidence that pathological conformation itself of the native protein drives the neuronal susceptibility to the aggregation. That it's really that conformation, that strain of the alpha-synuclein it has taken, which is the key driver why the certain cells are susceptible to multiple system atrophies conformation and not to the Parkinson's disease Lewy body conformation. But what is unresolved, what we do not know, what is the transcriptional impact of misfolded protein aggregation? And if we did know the transcriptional impact, could that take us closer to understanding the mechanisms behind the cellular and regional vulnerability? So for that, we used a spatial transcriptomics pipeline. We have included 8 Parkinson's disease cases, 4 MSA SND cases, 4 MSA OPCA cases, 8 controls. The pipeline we used was the nanostring geomics pipeline, which enables you to get the information for just over 18,000 genes. But then typically, as also in our case, after thorough quality control data processing, we ended up of having just less than 6,000 genes, which we considered reliable and interpretable. And what we see in these inferior olivary nucleus neurons, that both in MSA and to way lesser extent in PD, there is only downregulation of certain genes going on and no upregulation of any genes whatsoever when compared to the controls. And the biological relevance, these genes are very much involved in synaptic transmission, vesicle and protein regulation, calcium signaling, structural axonal transport, and metabolism. Then comparing the MSA different subtypes, to MSA SND versus Parkinson's disease, in this case, MSA OPCA versus Parkinson's disease. Again, we see that there is a heavy downregulation specifically in the MSA cases compared to the Parkinson's disease. And the directionality, looking at the both significantly downregulated and also non-significantly downregulated or upregulated genes, it's exactly the same. So what we are seeing here, that MSA SND and MSA OPCA cases in the inferior olivary nucleus do not differ in their gene expression profiles as a response to the alpha-synuclein pathology. And again, the genes involved, which are significantly downregulated, is pretty much the same as compared to the controls, which is very much involved in synaptic transmission, metabolism, calcium signaling, vesicle dynamics, and so forth. And it's kind of, for us, was quite surprising that that there was no difference really between MSA SND and MSA OPCA cases. And for that reason, we went back to detailed pathology review to really understand more why is that. And the reason for that is that actually it's the neuronal atrophy, but not alpha-synuclein burden, which correlates with the phenotype, specifically in the inferior olivary nucleus regions. So here, of course, as expected in Parkinson's disease cases, compared to any other MSA phenotypes, there's way less neuronal pathology or neuronal atrophy for that matter. But when we look at the neuronal alpha-synuclein inclusions in ION nucleus, as you can see, there is really no statistical difference in the frequency of these inclusions between the MSA OPCA or MSA SND cases. It's only the cases where there's a mixed combined OPCA and SND involvement where there's marginally more alpha-synuclein pathology. But the neuronal atrophy, however, is the one thing which really correlates with the MSA OPCA clinical and pathological phenotype. So it's the neuronal atrophy rather than neuronal alpha-synuclein inclusions, which kind of, on hindsight, explains that that's the reason why there is no differences in the transcriptional profile between the MSA SND and MSA OPCA cases. Another important thing to note is that neuronal alpha-synuclein alpha-synuclein pathology increases with this disease duration only in Parkinson's disease cases, but it's not the driver. The duration of the disease is not influencing the neuronal alpha-synuclein pathology in MSA cases. And what really this finding shows is that it is the pathological MSA conformation which drives the vulnerability to alpha-synuclein neuronal pathology, and what's happening as a cause, as a consequence of the misfolded protein aggregation, there is a complete downregulation, complete shutdown of major housekeeping functions for neuronal functions such as axonal transfer, synaptic transmission, vesicle dynamic signaling, really saying that these neurons literally have been destined to death. They're still there, but they're completely non-functioning for any kind of key neuronal maintenance functions. In the Parkinson's disease, obviously, as I showed before, there is very minimal, in majority of cases, no Lewy body pathology whatsoever, but it's really that disease duration, which to some extent also correlates with increased alpha-synuclein pathology, which is in contrary to multiple system atrophy cases. But at the gene transcription level, we do see subtle alterations, which very much are involved in proteostasis, oxidative stress, immune modulation, really kind of showing that probably these patients with a long disease duration, they therefore have higher alpha-synuclein pathology eventually because of the proteostasis failure, which this gene transcription profile tells is going on in the neurons where at that moment in time, there is actually no Lewy body pathology whatsoever. The other important thing to note is that by reviewing these pathology cases, close to 200, both MSA and PD cases, as I mentioned before, first of all, we extremely rarely see Lewy body pathology in PD cases whatsoever. And when we do see, it's very, in most instances, is the Lewy body morphology. However, what we have come across, there is a subset of PD cases with ION neuronal pathology, which histologically, it looks like MSA type morphology, which again very much suggests could there be a hybrid fold in this subset of PD cases. And at this moment of time, it would be absolutely amazing to do cryo-EM and to confirm whether it's the PD fold, the Lewy body fold, or MSA fold. But obviously, because the inclusions are so rare, and for cryo-EM at this moment in time, we really need grams of tissue to actually extract the filaments. That's not possible. But with the technology advancement, hopefully we will be able to understand more whether in a subset of PD cases, there is also hybrid MSA fold. So moving on about the digital pathology, how we are using that for MSA classification and characterization, it's very much to do with the co-pathologies. Why co-pathologies are relevant? They're relevant because of the fact that to interpret accurately the biomarkers, including the seeding aggregation assay, we need to know what's the frequency of co-pathologies in MSA cases. Once there will be disease-modifying treatment, obviously the presence of co-pathologies will influence the treatment response. And also, again, perhaps it's the co-pathologies which really make the phenotypic diversity and the regional vulnerability rather than the alpha-synuclein itself. And therefore, there might be that might be the driver of the phenotypic diversity and the atypical symptoms one can see in MSA patients. And about the co-pathologies, the important thing is that some of them are due to shared genetic environmental risks or synergistic interactions, but others more likely are explained simply by advancing age. And also importantly, some co-pathologies are relevant, in particular where they are sort of high load, high stages, but not all co-pathologies are relevant. And one really kind of therefore needs to study and understand in MSA cases, yes, there might be a co-pathology such as Alzheimer's type pathology, but is it actually relevant to that patient or not? And so the three key misfolded protein co-pathologies in general would be Alzheimer's type pathology, Lewy body pathology, and limbic TDP-43 pathology. So, and what we see in the Queen Square Brain Bank cohort, which again contains many MSA cases, we see that actually if we look specifically at the amyloid beta pathology, and here using a sort of a standard tau phasing, which consists of 5 phases, 0 being none, 5 being the most regionally advanced. So what we see here that in MSA cases who are ApoE3 genotype carriers, their amyloid beta pathology is way lower than PDD, Parkinson's disease dementia, Parkinson's disease cases, and even lower than control cases. And it's really only increased and similar to control cases if the patient is ApoE4 carriers, really highlighting it's the genetic risk which drives the amyloid beta pathology in MSA cases, and it's not any synergistic interaction with the MSA pathology as such. And if we again here look specifically in the SND OPCA or mixed SND OPCA phenotypes, again you can beautifully see that it's really the ApoE4 genotype which is associated with higher amyloid beta pathology. However, that ApoE4 genotype A beta pathology is not higher what we would expect in a neurotypical case. And for the similar stories also for the BRCA and BRCA phosphorylated tau pathology, that there is no indication whatsoever that in MSA cases there would be higher Alzheimer's type neurofibrillary tangle pathology in the medial temporal lobe regions. That if anything, again, in ApoE3 carriers, it's lower than any of the comparison groups, and also for majority on ApoE4 comparison groups, it's lower than in controls or PD or PDD cases. And here looking again in the SND OPCA and mixed SND OPCA cases, you can see there is no difference whatsoever. So it's not a reason for the phenotypic diversity in MSA cases. The Lewy body pathology, again, very, I guess, disappointingly, is not a feature of MSA and is seen in very rare cases, and obviously does not correlate with ApoE4 genotype, well, not necessarily obviously, but yeah, it does not correlate with ApoE4 genotype, and again, is not different based on the clinical or pathological phenotype. And the limbic TDP43 pathology, in our large cohort, we have a single elderly case, MSA patient with limbic TDP43 pathology, and vast majority of cases have no TDP43 pathology whatsoever. And again, in a typical neurotypical control case scenario, MSA TDP43 pathology, we see in patients over 70 years, and this patient is exactly exceeding the 70-year cutoff. So again, most likely it's the aging which has driven the TDP43 pathology in this single case out of 200 rather than MSA pathology as such. So it's really the aging and genetic risk which drives misfolded protein pathology development in MSA cases, which is in no way different from any other control case. What is, however, interesting that we do see T lymphocyte increase in MSA cases in substantia nigra. So here comparing a range of different diseases and the controls, there is, as you can see in substantia nigra, quite a big variability of the frequency of T lymphocytes, and it's not therefore statistically significant control from controls, although there is some trend. But what we do see that comparing specifically with the alpha-synuclein pathology, which is assessed using our quantitative pathology pipeline, we can clearly see that there is an obvious correlation between increased number of CD3 positive T lymphocyte presence in substantia nigra and alpha-synuclein pathology load, total alpha-synuclein pathology load in the substantia nigra. The reasons for that, we do not know what they are. So what are the key findings, and what I've went through today is that misfolded protein co-pathologies are uncommon and are explained by either advancing age or genetic risk, specifically ApoE4. T lymphocyte presence correlates positively with misfolded alpha-synuclein pathology in substantia nigra, and the work in progress we are carrying out is to investigate if the relationship between inflammatory cell presence, regional pathology burden, and clinical relevance can give us some cues about the causality and consequence. pathological MSA conformation is the main key driver of neuronal vulnerability to protein aggregation. And what's happening at the transcription level, there is a complete downregulation of major housekeeping functions in these neurons, which is a consequence of misfolded protein aggregation, really showing that these neurons are really not suitable for analysis to find out the clues of the cellular vulnerability in this scenario. And the work in progress we are carrying right now is characterizing the surrounding glial cells, astrocytes, oligodendrocytes, microglia transcriptomic profiles in the same regions. There are no differences in filament structure or seeding characteristics between clinical phenotypes, and the work in progress we are carrying right now is to analyze the regional oligomeric and aggregated alpha-synuclein and seeding differences across MSA phenotypes stratified by disease duration to really see whether these kind of earlier forms before the aggregates, whether those might explain or provide us some clues why there is the phenotypic diversity in MSA patients. And the work in progress I'm not going to talk about today, but we are very excited to finally have got funding to carry out, is we are developing nanopore-based profiling of synuclein post-translational modification, which we hope will be the future diagnostic marker as an added repertoire to the existing ones. And on this note, I will finish, but before I do that, I really would like to say a big thanks to my team at Queen Square Brain Bank, Yao, who is behind developing all the machine learning-based digital pathology quantification algorithms, Hemans, Fernanda, Toby, Nancy, who have really put their hearts in on making the digital quantitative pathology and spatial transcriptomics work to happen. Cyrus, who is an MSc student currently with us, working on the T-cell characterization in MSA and other disease cases, and also Patrick Cullinan, who couldn't be here today, but he's really the driving force behind the cell seeding experiments I've showed you, and also leading on developing, together with MSc student Mariam, the nanopore sensing. And very happy to take questions after.
Thank you.
Good afternoon, everyone. Thank you very much for the introduction. I'm really delighted to be here and to present some of the findings from my PhD project, which I'm very grateful to the MSA Trust for funding. So today I'm going to be telling you all a little bit about how we approach studying MSA genetics. And I think it's fair to say that there really isn't a clear role for genetics in MSA as of yet. The vast majority of the studies currently focus on germline variants, and we've had very few GWAS hits, and there are no apparent familial trends associated with this disease. But what if there are actually genetic mutations that are acquired later that could be at play? And these are so-called somatic mutations, which our lab is particularly interested in exploring. So somatic mutations can really occur at any stage during life, from early development to throughout aging and throughout the entire body. So the brain is no exception to that. And they can be small, like single nucleotide variants or indels, or they could be larger structural variants or even chromosomal abnormalities. And so you end up with an organ like the brain, which is actually a mosaic made up of distinct genetic identities that can lead to different regional and cellular vulnerability. And so as genetic techniques have really expanded in the last few years, we're more easily able to study somatic mutations. And this has led to the recognition that they do, in fact, play a role in the onset and progression of several neurological disorders, including epilepsy, schizophrenia, and in neurodegeneration like Alzheimer's, ALS, and frontotemporal dementia, and synucleinopathies. And so I'm just going to start by telling you a little bit about the synucleinopathy work that's been done previously by the lab and really formed the basis for my PhD project. So when you're looking at somatic mutations, it's really much more cost effective if you target your research to specific genes of interest. And our lab is particularly interested in somatic copy number gains of the SNCA gene, which encodes alpha-synuclein. And so the hypothesis is really quite straightforward. If you have extra copies of the gene in certain cells and in certain brain regions, this can lead to pathological aggregation of the alpha-synuclein protein and may explain some sporadic cases of synucleinopathies. And this really came from the observation that if you have inherited copy number gains of this gene, you end up with Parkinson's disease. And these also do show some glial pathology as well. So we know that these cells are vulnerable to excess levels of alpha-synuclein. But really, the first paper that was exploring this at the somatic level came out in 2018. It focused primarily on Parkinson's disease and control brains, but also a handful of MSA cases. And it's really quite a classical technique that I'm going to be talking about a lot today called FISH, where you fluorescently label your genetic sequence of interest. In this image, it might be difficult to see, but there are 3 copies of the SNCA gene in red and 2 copies of the reference gene. And surrounding the cell, there are actually brown pigments. This is neuromelanin, and this is what is used to identify dopaminergic neurons in the substantia nigra, which is particularly vulnerable in Parkinson's disease. And what they found is that there are overall a higher percentage of these SNCA gains in the dopaminergic neurons of Parkinson's disease versus controls. But actually, the highest overall gains were found in 2 MSA cases. So this is really quite interesting, although too low to really make any definitive conclusions about MSA. Then expanding on this, they started to look at more regions like the cingulate cortex and also adapting the methods that you can combine it with immunofluorescence. You can get better cell marker classification. So in this example, again, we see 3 copies of the SNCA gene, and this cell expresses NUN, so this is a neuron, and they found that there were higher gains in the cingulate cortex of neurons in Parkinson's disease and MSA versus controls. But actually, in just a few limited experiments, they looked at oligodendrocyte markers and found that the highest gains were found in the MSA substantia nigra. And so we're really starting to see a pattern here that these gains correspond to affected cell types within the different synucleinopathies. And then more recently, this was dug deeper in MSA. So they started to look at the alpha-synuclein proteins. We get more disease-specific context, associated with the gains and the actual inclusions. They also looked at different MSA subtypes like the S&D and OPCA subtype and the different affected regions. And what they found is that in a cell that had a gain, you had an increased relative risk for developing an inclusion in the affected brain regions. And not only that, but there were higher levels of these gains in the regions preferentially affected in the different subtypes. So that would be the putamen in the S&D subtypes and the cerebellum in the OPCA subtypes. But really, after all of this work, we still have some questions that remain. The first one is, are the gains that are occurring in MSA, are they occurring mostly in oligodendrocytes, which as we know is the main, but not the only affected cell type in MSA? What about copy number losses of SNCA, and what can that actually tell us about the disease? And lastly, how do we try and disentangle whether or not these gains occurred prior to disease, actually drove the disease rather than being a consequence of the disease progression? And so the first thing that I had to do was adapt the protocol. So I first adapted it to work on isolated nuclei rather than tissue sections. And the main reason for that is I can't call losses if I use tissue sections because I'll never be sure if I've actually cut through the nucleus. So if I actually isolate the nuclei instead, I can drop them on and I don't have to worry about the problem. So I did this for a mixture of S&D and OPCA cases as well as healthy controls. Healthy controls had not actually been looked at for these regions before. So this is from 3 brain regions, the putamen, the substantia nigra, and the cerebellum. And as I kind of briefly touched on, the rationale behind this is that the putamen preferentially affected in S&D, cerebellum is preferentially affected in OPCA, and the substantia nigra represents a commonly affected brain region. So once I've done that and I've dropped the nuclei onto a slide, I proceed with the FISH hybridization. It's using this custom designed oligonucleotide probe, which is about 50 kilobase in length, so targets about half the gene. And then I proceed with immunostaining. So I combine using an oligodendrocyte marker called SOX10 and alpha-synuclein to visualize the aggregates. And so here's what that actually looks like. So kind of on the top panel, you can see we have copy number 3 of SNCA, which is in red. In the middle, copy number 2, and at the bottom, copy number 1. And in green, you can very clearly see the alpha-synuclein inclusions, which are very well retained after nuclear preparation, and the SOX10, which is the oligodendrocyte marker. And so the results that I'm going to be showing you today are all unpublished, and they're from a data set which includes over 9,000 cells from 15 controls, 13 S&D, and 12 OPCA, for which the majority of them have all 3 brain regions analyzed. And so the first thing I'm going to show you are the oligodendrocyte SNCA gains. And so this is broken up across the 3 different brain regions. Each color represents one group. So OPCA is in orange, S&D is in purple, and controls are in blue. And what you can consistently see across all of the brain regions are that both MSA subtypes are higher than controls. And not only that, if we look at regional differences within each subtype, we find that there is a significant difference between the preferentially affected region and the lesser affected region. So this is the OPCA cerebellum versus putamen, and the S&D putamen versus cerebellum. When we look at the same thing in losses, we do also see that they are higher in MSA than controls, but they do not seem to show the same kind of regional predilections. And at the single cell level, we looked at relative risk. So you take the proportion of cells that have copy number gains, you calculate how many of those have inclusions versus copy number 2. And essentially, you are twice as likely of having an inclusion if you have an oligodendrocyte gain. This is only true when we look at the most affected brain regions and the common affected brain region, but not in the least affected brain region. As for losses, this shows really quite unclear patterns of relative risk. So we don't see it in the least or most affected regions, but we do start to see something there in the substantia nigra. And so we're currently trying to interpret what it actually means to have an SNCA loss. There hasn't been a lot of research into loss of function SNCA variants. So one thing we're kind of exploring is whether or not this is a secondary phenomenon associated with DNA damage. So we're looking at various DNA damage markers, as well as apoptosis assays, because that could also compromise the DNA. And then this is more of like a technical note, but we also looked in a subset of experiments at a reference probe, and we can still see that the significant difference between MSA and controls is only found with the SNCA probe. So we can see that for both gains and losses, whereas the reference probe is quite stable between both groups. And so here are some interesting correlations that we find associated with oligodendrocyte SNCA gains. So in the most affected brain region, the higher gains that you have leads to the higher inclusions, and this was significant. We saw this to a lesser extent in the substantia nigra, and it was completely absent from the least affected brain region. Not only that, but the oligodendrocyte gains correlate with gains in other cell types. And this is interesting because, as we know, they're not the only affected cell type. Neurons are also quite important. And so this could be an indicator that both are very important to the disease. And also, if this was an earlier clonal event, we might expect to see that they could be shared across multiple cell types. So this is very loosely supportive of that and not definitive by any means. And then lastly, the kind of average overall amount of oligodendrocyte gains negatively and significantly correlates with a younger disease age of onset. And so we think this could be supportive that they are driving the disease and may modify the severity of the disease. But lastly, we still really need to understand the timing of when this happened. How do we try and solve the cause versus consequence question? And the only chance that we really have at doing that is trying to capture the breakpoints of these copy number variants. But in order to do that, we need to do that with sequencing. We need long read sequencing because they're better for structural variants. And we need high coverage to pick up these low level events. But this is something that is really expensive to do on a whole genome level. And so we have to take a targeted approach. And so essentially what we do is we have bulk extracted DNA from 3 to 4 brain regions. We've got about 8 MSA brains and 3 healthy controls. We use a custom designed twist panel, which basically is focused on this 10 megabase region encompassing SNCA. The reason why we chose this is because that's where the inherited copy number variants frequently occur. We've also got some other synucleinopathy relevant genes on this panel. And then The probes essentially pull out your regions of interest, you PCR amplify, and then you sequence. So we did that on PacBio Revio, which gives you the most data output. So we get about 200 to 500x coverage across our region. And this is per sample as well. So we do think this is enough coverage to actually identify them. So all the sequencing has been done, but unfortunately all the analysis is still underway. And then kind of lastly, what we're doing as well is we're doing a smaller but interesting experiment where we want to look at genome-wide copy number variants. So we've sorted single nuclei based on SOX10 positivity, so our oligodendrocytes. And you can also see that we have this unique alpha-synuclein population that we can sort for. So we're actually getting the cells with the aggregates, and this is not present in the control donors. And so we're going to do single cell whole genome sequencing on that using a very well-established protocol by our lab to look at large-scale megabase changes. So that is essentially the end of my talk. Just some take-home messages. I think I've shown that somatic gains of SNCA and oligodendrocytes are correlated with alpha-synuclein inclusions at the single cell and regional level, supporting a role in pathogenesis. This could be either to the initial development of the aggregation, or they may modify the severity of the disease. Losses, on the other hand, show quite unclear patterns. We're still trying to really know what this means, and we are doing follow-up experiments on that. And then targeted long-read sequencing might actually allow us to characterize the CNV breakpoints. We can better understand how they arose, looking at the mechanisms and looking at the clonality of them as well. And so I'm just going to end there by thanking everybody from my lab, especially Christos, who's an amazing supervisor, to Zana, who's my secondary supervisor, our collaborator Fritz Sedlacek at the Baylor College of Medicine, to our funders, in particular the MSA Trust, and lastly to all the individuals at Queen Square Brain Bank that donated their brains, because all of this work would not be possible without them. So thank you so much for all your attention.
East, we have Chris. There's more young blood, but this is switching to the imaging format. So
Yes.
Thank you, Chris.
Thank you very much. Excellent. And thank you very much for the kind invitation. So in the next 15 minutes or less, I'm going to talk to you about brain imaging in MSA. And essentially, it's going to be a story in 3 parts. I'm going to attempt to introduce as to where the problem is at the moment and the limitations in trying to use MRI for MSA. I'll then go on to explain, well, why has that problem emerged and some of the approaches that we've developed to try and tackle that. And then I'll end by giving an example of the technology applied directly to an MSA cohort. So as have already been introduced by my previous colleagues, MSA is a variable disease. There's 2 broad phenotypes, MSA-P and C, and then a mixture in between. I think it's a relatively uncontroversial statement to say brainstem involvement is a common feature across the whole spectrum, particularly in the substantia nigra. But it's also, it doesn't affect just the whole brainstem, but it's quite tactical and focal. There are certain nuclei that are discretely affected, such as dorsal motor nucleus of the vagus, medullary raphe nuclei, pontine nuclei, and so forth. Knowing this and knowing that there are areas that should be affected, when we turn to the MRI imaging and we do a meta-analysis, what we actually see when we aggregate the voxel-based morphometry analyses is, yes, you're starting to see some changes, and I've updated this from 2 years ago, whereas previously it was only this spot just here. You're starting to get some basal ganglia involvement, but worryingly, the areas that we're most interested in, namely nigra, medulla, pons, nothing. Right? And you can go to lots and lots of different studies, and you actually have to take quite specialist approaches to start pulling that out. And for me, that's a major problem, right? And it comes down to, what do you expect this technology to do? And for me, the first and most important thing of MR imaging is it should be able to recapitulate the histological hallmarks of disease. These areas are affected and affected severely, and if we can't map it in vivo, we have a problem. And that's what underwrites all the other properties of MR, namely, you want reproducible biomarkers, you want to be able to map and track disease before signs emerge. But unless you have confidence that you can do this first thing, it all falls down. And we know we can do it. So in Alzheimer's disease, it's been very successful. You don't need many subjects to be able to map the medial temporal lobe atrophy. That's the hallmark of Alzheimer's, to the extent that this now forms part of the diagnostic guidelines for this condition. So why does MRI fail in MSA and other Parkinsonian disorders? Well, it all boils down to how we choose to analyze and measure anatomy in vivo. And it's a relatively simple technique. Doesn't matter what your style of imaging is or how you choose to do it. Ultimately, what you're trying to do is line up like-for-like structures and then pull a statistical handle. Yeah, whatever your test is or multivariate test is, it hinges on that simple fact you've got to line things up. If you can't line things up, you're going to encounter problems. The way we do it in imaging is we use either techniques called segmentation to identify common tissue types, or we use parcellation, so people will have pipelines to separate out different areas of the brain, but they're a related family of methods for various reasons. The brainstem is a beautiful and rich structure. There's around 138 different nuclei and 20,000 millimeter cubes of tissue. It's very evolutionary preserved. And yet, when we go to our common imaging techniques, this is what you get out. So this is free surfer, this is SPM. It divides it up into 3 regions at most, 1 region at least. Yeah, so you're never going to find anything. Yeah, and the consequence is when you're lining up your data, you're just introducing lots and lots of uncertainty and noise to the extent you never are able to detect your signal above that noise. These techniques have emerged because we use structural MRI and T1 weighted MRI, which is fundamentally just a photo of the brain. Yeah, the pixels the pixel values will vary a lot day to day, scanner to scanner, hour to hour. And it means you can only really analyze, they're designed for visual diagnostics. It means you can only really analyze gross shape properties. And so for the remaining of this talk, I'm going to be discussing more what's a type of MRI called quantitative MRI, which is essentially the same idea, but instead we've optimized them and we use a few extra acquisitions to be able to take out those non-biological things we're not interested in and actually go from a point where we have MR sequences where we can analyze the actual voxels and they have some meaning at a biological level. So, for example, R2 star and QSM reflects iron levels within the tissue, whereas things like magnetization transfer, it's the level of myelin, and so forth. And what's nice about quantitative MRI? Well, it gives you more precise measurements across scanners and centers. You have greater biological interpretability. It is more sensitive to pathological processes. And importantly, we can use this to be able to better map brain anatomy. And so the way we do this, and over the last 10 years or so, we've been developing methods to do this using quantitative MRI. And again, it's built off a simpler idea. Essentially, we combine 2 different types of images that have complementary but almost orthogonal types of information, and we combine those to be able to map boundaries that we wouldn't normally be able to see. So in this earlier work, we used magnetization transfer, which is myelin, and proton density, which is what it sounds like, to create maps to allow us to map the brainstem in vivo. We can see when we overlay our new tissue probability maps onto an ex vivo atlas of the brainstem, they seem to align quite well. But actually, we can take this now and plug this framework into the existing imaging pipelines and use that to then perform these analyses that we were interested in. Just to talk you through, so if you take the output of this, so these are Jacobian determinant maps, these are the things that ultimately you use to analyze at a voxel-wise level changes in volume. What you see with the standard approach is it's almost like you've been drawing with a pencil and someone smudged it. You don't have any information there in order to be able to make any inferences. Whereas if you use an optimized pipeline with dubbed quantitative segmentation, then you suddenly capture all this very rich information about local regional volume changes in different nuclei. And we did this in the past, and the first thing that we try to do when you've got large data sets, the simplest analysis you can do is, well, let's look at aging. Got that variable for everyone. And what we found with an aging analysis is that actually as people get older, you get midbrain atrophy. That was known. But the area that actually atrophy is causing this is situated just behind the substantia nigra. It's this area here, which because we've got other quantitative maps, we can ask the question, what's driving that reduction in volume? And in the center of that reduction in volume, you've got fall in myelin levels. And if you see those in tractography, you can show that actually it's a very focal loss of your cerebellothalamic tract that drives the midbrain atrophy you see with normal aging. And that's what's nice about having the quantitative MRI techniques is you have this broader repertoire of images and modalities in order to be able to understand what's driving the changes in your data. So what happens when we apply it to MSA? So I have to thank Tom Fultony for this opportunity. Actually, he was running the exenatide in MSA trial, which was a really unique opportunity to actually at the same time bring some of his participants across the road to where I work at the Phil and do our MRI acquisitions with this. It was paired with an ongoing study that I'm running in Parkinson's called QMAP-PD, where we already had existing data for healthy controls. These details are probably of less interest, but just very briefly, we had 32 MSA participants and 84 controls. The analysis I'm going to present is just a two sample t-test. I wanted to ask the question, where are the differences in MSA versus controls? And can we map the histological hallmarks in vivo? The data I'm going to present is what's called a multiple parametric mapping protocol. It's 800 micron. It gives us 5 quantitative images. We use the magnetization transfer and proton density to do our quantitative segmentation, which really means we go from having 2 tissue classes to 8 that we use for the analyses. I'll walk you through the results, but really I'm going to present the standard approach you would get with just using SPM out of the box, the new approach, and then using this new approach to drive down the amount of smoothing we're doing, which gives us greater spatial sensitivity. So this is using standard VBM out of the box. You'd be very pleased if you got this in your imaging analysis. It's quite good. So this is 6 millimeters of smoothing, 2 tissue classes. You can see you've got quite a lot of cerebellar atrophy. You've got some striatal atrophy. These are mixed groups. I haven't tried to separate out MSA, CEP. We just put them all together for this first analysis. What happens when we up our number of tissue classes and rerun exactly the same analysis with exactly the same amount of smoothing? Well, suddenly we start seeing more spatial sensitivity already. So you're now getting the cerebellar white matter atrophy that's driving it. You're certainly starting to see some brainstem changes in areas that you would be interested in. But really, it starts to pop when you drive down this, the amount of smoothing, because these segmentations are designed for fine-grained analyses. And all of a sudden, your effect size, so your T statistic is dramatically greater. You're suddenly starting to get areas that you're interested in. So, for example, in the medulla, there's the medullary serotonergic group, the dorsal motor nucleus of the vagus. That small spot there going down the fourth ventricle is your locus coeruleus. We have our cerebellar thalamic tract and substantia nigra here. In the thalamus, there's the pulvinar and medial dorsal nuclei, and you're even starting to capture some primary motor cortex changes there. So these are very focal and very precise. What's actually happening if you pull out the results, just to walk you through it, is you go from your classic position, where if you just do the same analysis with the increased tissue classes, you get a lot more results, as you'd expect. So it's about 20% more. But actually, when you drive down the amount of smoothing, the proportion of results surviving this family-wise error correction increases, and your overall T statistic is much greater. So the take home from this is that basically your effect size is bigger, and you're going to need less people in a sample size in order to detect an effect. Because we have the rest of our MPM images, our quantitative MRI, we can then analyze each of those in turn to see if we can understand what's driving these changes in volume. And what we find in the cerebellum is we get widespread and pretty profound loss of myelin, which marries up with the post-mortem literature. The R1 is a bit of a mix of iron and myelin. It's less dramatic. You'll start noticing there's a little bit of artifact just around the base of the brain there. That tends to happen in the MPM images. It just needs a bit of skull stripping to get rid of. The proton density, so interestingly, the proton density goes down in MSA. So often in tissue damage, you see more protons circulating around that can go up. Looking at the MS literature, it's likely that actually reflects a more chronic degree of damage to those regions. Our R2 star measure of iron, we get very focal increases in iron within the putamen, pallidum, and also the substantia nigra. You get corresponding decreases in the middle cerebellar peduncle, but that's because iron co-locates with oligodendrocytes, and so often you do see that in areas where you're losing myelin. And then finally, the QSM, so quantitative susceptibility mapping, we did expect to see a bit more, but actually it wasn't as dramatic or as compelling as the R2 star data. And I think there are various reasons for that. QSM is quite a finicky modality. Interesting, you see an increase rather than decrease in signal in the middle cerebellar peduncle, which has been associated in MS with chronic inflammatory responses. So to summarize those results, that's very quickly going through a very large range of results. The key take-homes are that you get profound volume loss, particularly in the cerebellum, brainstem nuclei, pallidum, and nigra in areas that you expect to see. You get particularly severe loss of myelin, particularly in the cerebellum and the cerebellar white matter. But interestingly and uniquely, it also follows other tracts. So this area here is your corticospinal tract, which you're just recreating simply using a voxel-wise analysis. And this is best mapped using magnetization transfer imaging. And then your pallidal and nigral volume loss is associated with focal increases in iron. And that is best mapped using the R2 star images. So to conclude, I started off by stating what the problems were with the standard MRI approaches and what we're missing. I explained that the reason we're missing this is because essentially we are unable to map it, and I have outlined an approach using quantitative MRI that allows us to map these structures. And I presented a QMRI-based in vivo histology in the MSA cohorts. What I haven't had time to talk about, and one of the things we're pushing forward, this is a group level analysis. We're building off the same technology. We can now drive it down and do single subject-based mapping to see how far your volumes and your regional volumes deviate from normal. And that's something over the next 3 years we're very excited to take forward. I'd like to thank, there's many more people I could thank than I could possibly fit on. First and foremost, all of the participants. People give their time freely. We rely entirely on volunteers. None of the work is possible without them. I've had a fantastic group over at the field, particularly Charlotte Dorr, who is amazing at getting people into the scanner, is very patient, and we can do a lot of positioning. MSA cohorts, if you've ever tried to scan and study them, they're extremely complicated, very difficult, and you need a really dedicated team around you to really help support the individual. And of course, Tom and his ex-Xenotide and MSA team. That's the end of my talk. Thank you very much. Happy to take that.
Welcome back to the afternoon session. My name is Chris Kobolewski. I'm a consultant neurologist in Salford and chair of the scientific advisory panel of the MSA Trust. So let's get started. So I'd like to welcome to do the first talk someone who, if you know the MSA Trust, doesn't really need any introduction, Karen Walker. So Karen Walker has been the CEO of the Multiple System Atrophy Trust since November 2015, and she's done a fantastic job during that time. And I think we all very much appreciate her work, her tireless work on behalf of people with MSA. So without further ado, I'm going to hand over to Karen to tell us about shaping MSA Trust strategy. Thank you.
Thank you very much. I just want to very briefly say welcome back after the afternoon session. I'm sorry you've got me for the graveyard slot, but hopefully I won't bore you too long, because really this has been all about the research, and we've had an amazing morning, and I just want to add a little personal thank you to all the amazing speakers we've had, because they've done such a wonderful job. And we've got more to come this afternoon, which is even better. But I wanted to just to talk a little bit about MSA Trust and where our strategy has taken us. This is we're currently into our third strategy since I first came on board. It kind of went a little bit adrift during COVID, but we got through it well, and we have an amazing team of volunteers in our trustees and our scientific advisory panel, chaired expertly by Chris, for which we're very thankful. And so when he says about shaping strategy, he has also helped us enormously to shape our research strategy. And so we have our overall strategy and then our research strategy. And this year, what we have done for the last two strategies is actually to use the voice of people with MSA. And so we have put out 2 needs surveys. And later, just after me, I believe Professor Schrag is going to talk a little bit about those and give a little bit more detail. And so I wanted my talk not to be a talk, but just to be a film. It's a 7-minute film, and it's a compilation of people who have MSA and their families, and some of the key difficulties that led us to think differently about the services we provided. And I know earlier today someone mentioned the fact that we looked at the emotional side. So now we have a counselling service, and we're actually looking at the moment, we're developing that into group counselling as well as individual. And we also have now counselling for carers. So that was a very valuable thing to realise people needed. And of course, the support groups that we run, we have built and developed and grown further when people started to talk about the carers needing time together and being able to talk openly with people that know about this condition. So we then have hopefully tried our best to meet that need as well. So we've done what we can, but we're always open to new thoughts and ideas. And we will go back out to our people with MSA and their families and their carers in the not too distant future with our next questionnaire asking them. But we just have to be careful that we don't push them into questionnaire fatigue, survey fatigue. That's not, we have to be so careful of that because there's a lot of the research that we know the researchers need. So we don't want to take up too much time for our people with MSA. So anyway, I shall play the film and obviously really happy to talk to as many of you as possible afterwards. Thank you. I just wanted to finish by letting you know that, of course, we have sadly lost a couple of these people. And indeed, one of them has had their diagnosis changed again. And again, that sadly is all too common for people. So, but they all gave permission that even if they had passed away, that we could still use their details and use the films that they very, very kindly agreed to make while they were in that particular condition. Thank you.
Thank you, Karen. So really continuing the theme here of improving care and patient-centric approaches, which Karen has sort of very eloquently started off. So I'm going to invite now Professor Annette Schrag from UCL to come and talk on care needs in MSA and the results from the MSA Trust Needs Survey. So Professor Schrag's clinical researcher at UCL Institute of Neurology, Queen Square, around the corner from here. And she's produced numerous high-impact papers over the years on assessment and management of Parkinson's and other movement disorders, including MSA. So thank you, Annette.
Thank you very much to the MSA Trust for inviting me to talk about the two surveys that Karen has already mentioned. And thank you for this introduction and this video, which really reminds us what a dreadful disease this is. So as I said, this is work done by the MSA Trust, and a lot of people obviously inputted in this and went through various stages. So people from the MSA Trust, but also the I think steering board or committee. Many of whom you've already heard today. Also David Oliver, who's a palliative care consultant, and also Neil Quinn, who is you probably all know. And of course, very importantly, also people with MSA and their families contributed to the design of this survey. So there were two surveys, one in 2019 and one in 2022. They were slightly different. There were some questions that were just in one and not in the other. The questions that overlapped, so the majority produced very similar results. So I'm going to talk a little bit about both of these combined. The response rate was actually very good for a survey of this kind, particularly considering how disabled, of course, a lot of the respondents were and how busy their carers were with looking after them. So 22% in people with MSA, 29% of carers, and then also former carers where the person with MSA had already died. 40% of them filled in this questionnaire. So that really reflects how appreciative we're about the work of the MSA Trust. So in terms of age range and demographics, they, I think, are fairly representative for an MSA population. So the majority were in the 65 to 74 year range, a third and slightly younger, 55 to 64, and the others older than 74 years. Disease duration, a third had a relatively short disease duration of 5 years, but there were over half of them who had a disease duration of 6 to 10 years, so would have been really quite disabled at this point. So there isn't a measure of disease severity, obviously, in a questionnaire, but I think that is a correlate of that. 52% were male, and the vast majority were white respondents, and that's very typical, I think, of research, but also of engagement with charities. So I think that's very much as expected. In terms of the carers, 62% were female, and I think almost all of them were the spouse or partner of a person with MSA and had lived with them throughout the disease course. And again, the duration of disease was fairly similar, perhaps slightly later, obviously, in the disease stage. So there were 17% had the person with MSA who had already died had a disease duration of more than 10 years, which obviously is more than the mean, but of course, some people do, given that it's the mean, do live for longer. But there were, because it's a questionnaire study, and people were self-reporting their diagnosis, there is also the possibility and the likelihood that some of them did not have MSA and possibly were later re-diagnosed. Nevertheless, given that it's such a large survey, I think that it probably is representative. So as one might expect, the main symptoms were those that we're very familiar with: movement problems. All of them had some movement problems, autonomic features, and this encompassed really all of the ones we've talked about this morning, but also temperature control and sexual dysfunction. And then speaking, swallowing, breathing were combined in one. I think to all intents and purposes, this is probably mostly speaking problems, and with more severe problems swallowing. That wasn't differentiated. And interestingly, a lot of people also had reported sleep problems, perhaps not so unusual, but also pain and concentration problems. And these were fairly similar in men and women. So in terms of the severity of the symptoms, the ones that were reported that were severely affected or very affected by autonomic and movement problems, most of them reported they were severely affected by those. Difficulty with speaking, swallowing, breathing, about half of them. Less commonly severely affected by sleep problems, but still 31%. And a quarter were severely affected by pain, difficult concentrating, and of course also falls. And a small number also had feeding tube issues. But what was really interesting, above and beyond what these symptoms that we sort of always associate with MSA, there were a lot of mental health, emotional issues that Karen has already mentioned. So fatigue was universal, more or less, clearly multifactorial. Depressive symptoms also very common. So this is not necessarily major depressive disorder fulfilling specific DSM-5 or other criteria, but depressive symptoms, 70% reported anxiety and 75% stress. And all of these were slightly higher in women, which is also in the general population. These are also more common in women, but certainly in this population as well. But importantly, half of them felt that their mental health was were only partly, if at all, met, and 30% didn't feel they were at all met. And also the former carers reported that 41% of them felt that the emotional needs of the person with MSA who had already died had not been met. So this is clearly something that we probably ought, or somebody ought to pay more attention to, and this ought to be addressed. In the second survey, there was a little bit of exploration how this was met, and usually it was in informal ways and by the MSA Trust as well as family and friends. And of course, this is not surprising. The psychological strain for people with MSA as well as their carers is of course multifactorial, all the mobility problems, the autonomic problems, but also the impact on social activities, pursuing as came out in the video, pursuing the normal activities they used to enjoy, the social interactions, they all contribute to this psychological burden. In terms of what they reported having been provided with, so just very factual, the equipment that people were provided with, I think one has to say that this is possibly better than it is in the overall population of people with MSA, because these were all people, I believe, who were members of the MSA Trust or were in some way aware of this. So they would have been better informed and probably had better support than the average of the MSA population. So most of them had some sort of walking support. A third had a urinary catheter. 13% had some sort of communication aid, 9% ventilation support, and 5% a PEG, and 1% a tracheotomy. A third of them relied on someone else to help them walk. And more or less all of them had some input from professionals on mobility aids. But nevertheless, about 40% of the carers really desired more help with the fact that there is a lot of physical strength required and helping with the manual lifting and with the falls, which is clearly a big strain physically, and many had back pain and other health issues. And of course, low mood also in carers. And we know that from other disorders that depression in the person with the disease and the carer often correlate quite strongly. There's the social and financial impact. Perhaps again, not surprisingly, the social impact was was high and probably increasingly so in more advanced disease. Many felt socially isolated and their relationship with friends had deteriorated, and sometimes in a high proportion also with their partner. But in many it stayed the same, and in some it even improved. And perhaps not surprisingly, almost a very high proportion reported their sex life had been affected. And obviously that wasn't broken down further into physical and psychological features, but very few had received any treatment for this. There was also financial impact. Half of the respondents reported that their financial needs had not been met or only partly met. And the same was reported by the carers, former carers. And for a third, this financial impact continued after the person with MSA had died. So in the second survey, there was a question more specifically asking about whether the MSA impacts on enjoyment of things they used to do and doing new things. And again, this reflects that living with MSA is not just the actual physical symptoms of MSA, but it's mobility issues, concerns around toilet availability, concerns about being able to communicate, difficulties with just getting around, traveling, that all impacted on their ability to do things they used to do or even start new things. So how are we doing in terms of the provision of health service? This graph is really to show what was offered, what people had, what sort of input they had from health services, and also how happy and helpful they were with it and how helpful it was. So in blue, and in order, you can see most of them had, or almost all of them had, a neurologist, a Parkinson's specialist, and a GP. And then many had occupational therapy, physiotherapy, which is what we often refer to. Many had contact with a Parkinson's disease nurse, and then, of course, the MSA trust nurse, continence nurses, wheelchair services, and others. In terms of the satisfaction with this healthcare provision, it's interesting. So in absolute numbers, that was fairly similar, but in relative numbers, the satisfaction with these services was really highest for the palliative care services. And it's not on here because not so many people had it, but hospice care also had very high satisfaction. So, and that also came out in some of the other questions of more open-ended questions. So an MDT approach and a coordinated care approach was something that many wished for. And this was also a similar picture was seen in the survey of the former carers. So what other therapies did people find helpful? So many found massage and exercise helpful. And there were a number of other options in terms of alternative medicine and supplementary therapies. And they were really used by very few. And also the helpfulness was also not very high. So these are the two that people used and found helpful in a proportion, not in everyone. And only 26% had any sort of psychological help, such as counseling. And of those who had it, the vast majority found it helpful. So the other unmet need is not just the actual provision of services, but also information. And we heard about the delay in diagnosis, and that's something we've already talked about. There's a lot of effort to improve early diagnosis, and with new biomarkers, hopefully that will improve. But it's also at the time of diagnosis, also a time when there's a real need for information, and we're not that good at it, partly because there are so many other things to consider, and partly because we often think that when we communicate information, then that's enough. But we really need to provide some written information because people usually don't take in enough. And also, we can't communicate everything in the consultation. So that's really a need that I think is being met by the MSA Trust. But of course, not everyone is a member or in contact with the MSA Trust. And here's a list of the information needs. But importantly, I think for people, they want to understand what it is. They want to know about the prognosis. They want to be able to plan. Here, just very briefly, just expanding on the fact that care was also affected, of course, by this disease, and they would like to have more support for many of these aspects. I've already mentioned this. So just talking about the information need and the need to plan. This is, of course, something that is done very much in the realm of palliative care. But of course, palliative care, specialized palliative care, is not available to everyone. And also, we are not linked in a very effective way, usually, to palliative care services. So I just wanted to show us a few slides about this, because it is something that, to a certain degree, is just an extension of an MDT approach. So palliative care is nothing else as providing symptomatic treatment or treatment to alleviate symptoms. That's the meaning of palliative care. But importantly, I think in practice, it includes not just the physical aspects, but also the psychological, psychosocial, spiritual aspects. And it typically, in cancer care, concentrates on pain management, but it really is a symptomatic treatment. And that can include very specific treatment for individual symptoms. And with palliative care, we know this from Parkinson's disease, but of course also from other disorders, quality of life can be significantly improved. And importantly, in terms of planning ahead for the future, advanced care planning should also be in some way addressed within this. So in terms of palliative care, just briefly, what the survey showed, that a really high proportion had received some palliative care, and I think that's a higher proportion than we would have expected. But ongoing palliative care is of course something quite rare. But there were only very few people who wouldn't have wanted any palliative care involvement. So it is something that we sometimes shy away from, but should consider offering. And advanced care planning. So there's of course many aspects to this. In these surveys, a very high proportion had already made a will, some sort of proxy decision-making decisions. About 20% had made some advance directive or do not resuscitate order, and a proportion, but not very many, had a personalized end-of-life care plan. But it is important because people, even though they know it is a terminal disease, it shortens lifespan. It was quite a surprise in this survey to hear that only 20% or so expected the death at the time when it occurred. This is in the former carers. Only and in 37%, death was unexpected, and in the remaining 41%, they did expect death, but it was sooner than they thought, even when they were in hospital. So there is a need to really talk about this more openly and make plans earlier. And this is another result from both of the surveys. When people were asked, whether they had thought about what they would like to happen at the end, towards the end of life, and if so, whether they had discussed it with a healthcare professional. The vast majority had already thought about it, of course, but a quarter hadn't. They hadn't. They didn't want to engage in this thought. But only very few had actually discussed it with healthcare professionals. This is despite the fact that when they did have a conversation, the majority did find it a helpful conversation. And in terms of palliative care, the majority of people did want to have some sort of discussion about palliative care. Not everyone wanted it, but it is certainly a discussion that we ought to have at some point in the disease course, and probably in a disease like MSA, it should be earlier rather than later. And just in terms of addressing the information need and training need, I'm not aware of any of these massive online courses. There are some for MSA, but there are some for late stage Parkinson's disease, and many of the issues, of course, overlap. And this is one from the PD-PAL project, which is on the Future Learn platform. And just recently, an Australian initiative for Parkinson's MOOC, which also has some relevant information. So just to conclude, obviously, MSA is associated with a number of physical problems, but also psychological, social, and financial difficulties. And addressing these is really important for people with MSA and their carers. And I think the MSA Trust is doing a great job at addressing these, but we need to make sure this is something that is more available to everyone. An MDT approach is certainly something that is welcomed and experienced as beneficial by those who receive it, but also those who don't have it, who would like to have an MDT approach with more coordinated care. And ACP discussions is something that we really ought to get better at including. And I'm talking about myself as well, because it is such a difficult discussion to have, and there are time issues. But certainly something we need to tackle. And linking to specialist palliative care services is important, whatever the local availability is. It differs between different trusts, but certainly something that we should consider for all patients with MSA. So thank you.
Thank you very much, Professor Schreib, for that really comprehensive look at the MSA Trust Needs Survey. And we'll take some questions on this at the end of the session. So for the final talk, I'd like to invite Dr. Seema Sheikh to present. So Dr. Sheikh is a Senior Research Fellow in Neurology at the Wessex Neurological Centre in Southampton, and she works on a number of trials, including clinical work with Dr. Boyd Ghosh and Dr. Luke Massey, who run that clinic. And she's going to tell us today about variation in quality of life over the course of MSA and potential treatments. So thank you, Dr. Sheikh.
Thank you very much for that introduction. So I'm going to discuss quality of life in MSA, in particular determinants of quality of life, the questionnaires that are important, and potential treatments. And I have to say that this PowerPoint presentation has been put together with my consultant supervisor, Dr. Boyd Ghosh, at the back, and also with very good help. Thank you. So to begin with, I will discuss a systematic review of the literature that I carried out and the questionnaires that we formed from there. And then I will touch upon the Prospect UK study and the findings from there. And then go on to the treatments and practical approaches that I've come across in my literature search and review. So this is a quick overview of my methodology in our systematic review. And I'm sure everyone here is familiar with this approach with the PICO criteria, but essentially our search criteria was quality of life in multiple system atrophy. And this is a Prisma graph identifying the studies that we found from various search engines. And after screening and deduplication, we had 28 studies. And we divided those into those that focused on questionnaires on quality of life, those that focused on determinants, and those that affected on quality of life treatments. And this is a summary of the types of studies that we came across. So longitudinal and cross-sectional, as well as a few systematic reviews, which were really helpful for me to understand. And a range of quality of life questionnaires, and I will briefly talk about those. Other points to consider are the scales that we use in MSA, including the UMSARS. So the first questionnaire here is the European questionnaire, which is very common and used widely in regulatory bodies such as NICE. It has two parts. So the first part has 5 dimensions as listed here, and patients can rate their quality of life depending on whether they have no problems all the way up to, for example, in this particular domain, I'm unable to walk about. So from 1 to 5, and there's 5 dimensions here, so it results in a 5-digit number. And crucially, we have a visual analog scale here, which patients themselves can rate their quality of life from the best health they can imagine to the worst health they can imagine. And we've also looked at the fact that this has been validated by other questionnaires focusing on quality of life, such as, and within this framework, we've got the EQ-VAS, so that's validated its own questionnaire to show that there is significant positive correlation between the utility over here and the EQ-VAS scores. So all of these correlate well with the visual analog scale. Secondly, I've got the MSA quality of life questionnaire that Professor Schrag has put together. And this is a very comprehensive questionnaire. It has 40 items divided into motor, non-motor, and emotional domains. Crucially, this also has a visual analog scale measuring overall life satisfaction from worst to best. And each item is rated from 0 to 4, where 0 is no impairment and 4 is extreme impairment. So the higher the number, the worse your quality of life. And we've had a look at the literature, and indeed this questionnaire does measure life satisfaction. So the MSA quality of life subscales negatively correlate with the life satisfaction, the VAS score on the MSA quality of life. And it also correlates with the EQ-VAS in the European questionnaire. The motor subscale also correlates with motor severity in other questionnaires, such as the PDQ-39 and EQ-5D. The depression and anxiety have also been measured here, and that's been so emotional domain has been correlated with the HADS, the Hospital Anxiety Depression Scale, and also the EQ-5D and PDQ-39. And pain and non-motor features of so non-motor subscales in this questionnaire have also shown correlation. So our colleagues have also developed the quick MSA quality of life, which is essentially a shorter version. It has 15 items instead of the 40 items. So they do miss some items such as balance and urinary dysfunction, which we heard is very important. However, in this in this study, they did say that the quick MSA is an efficient tool for reducing respondent burden And I understand that patients with MSA and their carers are filling out questionnaires all the time, and this would be really helpful for clinical trials as well. This is the 36-item short form survey, which is also very common. It's not just used for MSA, it's widely used. It has 8 components. And as you can see here, it's quite comprehensive. Again, so in this scale, the higher scores indicate better health. And there's a physical component summary and a mental part as well. And we've also looked at the literature, and this questionnaire also shows that it is about quality of life and life satisfaction. So this SF36 subscales here do positively correlate with the visual analog scale in the European questionnaire. And however, there are parts such as the emotional and well-being part that didn't significantly correlate. So we have to be aware of that as well. Okay, so I'm sure many of you are aware of the Prospect UK study, but I'm just acknowledging Professor Hugh Morris, who led this. And there are many phases of this study, but essentially we had in our cohort that we analysed 164 patients with MSA, and there was MSAC and MSAP, so there were 85 MSAP patients and 64 MSAC. The mean age was around 59 years old, and mean duration was around 8 years. There was a pretty even split between male and female, and as you can see, survival only 34.7% still alive. Ethnicity-wise, white ethnicity was predominant, and various questionnaires were carried out, including the MSA Quality of Life questionnaire, but also caregiver burden questionnaires and disease severity questionnaires such as the UMSARS. So we found that the MSA Quality of Life total score is correlated to disease severity as measured by UMSARS. And that's UMSARS part 1 and part 2 separately, but also together. And you can see this is a significant correlation. And we're looking at particular items, and many of the items were significant, but these difficulty moving, walking, standing, and difficulty with housework came right at the top. Also, the UMSARS, the global disease severities part, also was significantly associated with item 11, difficulty dressing. But I have to emphasize there are a lot of significant correlations here. This demonstrates how we put the MSA items into 4 domains, like our French colleagues here. So motor, non-motor, oropharyngeal, and emotional. So 4 parts of the motor were brought into and made into the oropharyngeal domain, and 1 item 25 was not used or deleted because it was similar to another item 1 concentration. So we looked at the domains, and we noticed that the motor domain correlates most with UMSARS or disease severity. However, as you can see with the p-values here, that the oropharyngeal and emotional domains were also very significant. And that's important that we don't neglect those, because they can be forgotten, as we heard from Professor Schrag. We also found that MSAP patients have to give significantly lower quality of life than MSAC patients. And this was from the life satisfaction part of the MSA Quality of Life questionnaire. And our colleagues from Professor Mesnier team have also found that female patients experience a poorer overall health-related quality of life than males, and that a delay between symptom onset and diagnosis can also be associated with a worse quality of life. We also looked at some longitudinal aspects, and we found that the MSA-P patients do indeed seem to deteriorate more rapidly than patients with MSA type C. And you can see this in that the x-axis here, that's the baseline UMSAR score that the patients had. And on the y-axis is the rate of change in whatever domain of the questionnaire. So here we've got rate of change in the total MSA quality of life score. And we can see in the orange or red that the MSA-P patients, they deteriorate more rapidly than the MSA-C. And this is similarly significant for the emotional domain here. We also saw similar trends for motor and oropharyngeal. However, these weren't significant in our linear regression analysis. However, and putting all of this together, compared to MSA-C patients, MSA-P patients in other studies have shown higher oropharyngeal, non-motor and emotional and social deterioration, which is in keeping with our findings as well. Our colleagues have come up with this brilliant diagram, which I have stolen for our slide, to show that there are distinct patterns of deterioration throughout the disease course, and we can really use this to target treatment. So people with MSA may start off with problems with drooling, for example, and throughout the disease, they may have problems with fatigue and depression. And in the middle to late stages, they may have problems with the motor aspects, and which go on to their activities of daily living, such as getting dressed and using the toilet. So this is really a brilliant diagram here. And it also shows that pain and discomfort are usually more at the later stages. So putting that in the forefront, we can then direct the treatment. So in terms of drooling, there's lots out there, and I've seen, so for example, using atropine, eye drops taken orally, for example, and inhalers. Another thing to point out is that early referral to speech and language therapy is really very key, both to preserve the communication and also to keep our patients safe. And then, yes, so urinary dysfunction, as we've heard today, very important. And depending on the type, there's lots of interventions that we can do, which are quite easy to do if only we ask the patient and know their symptoms. And then in terms of stages 2 and 3, using mobility aids, such as were described earlier, with whether they're walking sticks or Zimmer frames, is really helpful in terms of quality of life and getting out and about as much as you can. And other complementary therapies, such as massage therapy, really helpful, and exercise. There's also inpatient rehabilitation. If we can have it, that would be brilliant. It significantly improves not only motor symptoms, but also the emotional and mental health aspects that we've seen that really do suffer just being around other people. And then palliative care. So this is crucial. Neurologists can play a huge role in managing symptoms here, but we can also be doing early referrals to palliative care and making sure patients know what palliative care is, because there is a misconception. It's also very important that we talk about advanced directives and give patients the autonomy that they deserve, and also just improving their access to whatever treatments that they would like or wouldn't like, and having that honest conversation early rather than later. And then there are other treatments in the research pipelines, for example, high cervical spinal cord stimulation, which has had some positive impact on speech quality. However, there is more research needed for its effects on gait. And throughout the disease course, we need to ensure that patients have a support network, whether that's their carers, whether that's their family, or even being there as clinicians. I've also understood that access to specialized care for patients is really beneficial. The fact that they know that clinicians know what they're talking about really helps. And then in terms of fatigue and sleep, so CPAP has been shown to be quite helpful, though some people may not get on with it, but on the whole, it's been helpful. Therapeutic education programs to ensure our patients know what this condition is, what to look out for, and also their carers know what to look out for. That's been shown to have potential benefits. And in terms of depression, for example, fluoxetine has shown potential. So in summary, we've gone through some of the quality of life questionnaires, and some are a bit longer than others, but it depends on the time that you have with the patient and also perhaps the research trial that you're undertaking. And key messages here are that MSA quality of life total score is correlated to the disease severity as measured by UMSARS, that MSAP patients seem to have a lower quality of life than MSAC patients from what we've found, and also a more rapidly deteriorating quality of life than MSAC patients. That female sex is also associated with a poorer quality of life. And motor is a key determinant of quality of life. However, we wouldn't want to neglect or forget the emotional aspects and the mental health aspects, and that there are distinct patterns of deterioration throughout the disease course, which leads to treatments every stage of the disease. There may be particular treatments that we should be getting our patients onto and almost preempting. So thank you. That's the end of my talk.
Thank you. Thank you very much. So I'd just like to invite the speakers from this session, if you would, to just come up to the table here, and we'll have some time for questions. I think we have hopefully some time. Okay, so would anyone like to start off with some questions? If you would please introduce yourself if you do start with a question. But Boyd, Dr. Ghosh, would you like to start things off? I don't know if we can get a microphone. Possibly, it might be maybe not. You might have to shout. Okay.
I think one of the striking things was that, at least in this population, the physical needs were all being addressed, whether obviously we know that we can't alleviate them completely, but they were being addressed in some way. But the emotional aspects were not addressed at all. So very few people had any psychological input or any way of this being addressed. So I think that is an important one. And the other thing I would say is the carers who don't. I think that everyone is entitled to a carer assessment, but we all know that carers often don't view themselves as carer and don't seek particular help for their caring tasks and for their own health, which is crucial, of course, as we know, for them to be able to support the person with MSA. And then the third thing I would say is the ACP, the Advanced Care Planning. And if people die unexpectedly with a disease like MSA, that's really, that tells us something about our communication. And of course, there's also, it's probably been mentioned at some point, but that's probably not enough. It is something that needs to be done on a more continual basis.
Thank you. Professor Bhatia.
I find that some of my patients find the word palliation or palliative they have a bit of an antipathy to this word, which sort of brings to them the inevitability of what's going to happen, and sort of brings it to. I just wonder. I think you mentioned a very good point there about how to. One of the things we need to learn is perhaps how to say the same thing, but in a different way, perhaps, or in a. But when you say, okay, have you been contacted by the palliative team, or would you like? And that really rankles with them. That really makes them upset, and perhaps even puts them off. And I just wonder whether you have any comments about how should one. It's the most very important aspect, but how do we go about avoiding the P word? Is something which I think deserves some comment, perhaps.
Yes, no, I find this. I find the same, and there are some palliative care services who avoid this term palliative care because it sounds to people sometimes like you're giving up on them. And I tend to talk about MDT and support and mention some specific things that are offered in the specific place that I'm referring them to. And that often works the best because then they know what to expect, or there's a specific reason to go there, even though it is a palliative care service. And also explaining our community. Yeah. So the other thing is, of course, just to make. So obviously in Parkinson's we have the same thing. So I was part of this PD pilot project, and there was a lot of drive to introduce a more standardized way of assessing the palliative care needs, but it was very long. So we did a very short version where there's effectively a 2-page thing that people can fill in at home, and then complete it with Parkinson's nurse or the MSA Trust at an in-person session, the more sensitive questions. So I think that is one way of how one could address it.
I think if I could maybe add, it'd be interesting to see what my colleagues on the panel think as well. I think two things. I think one thing is about, and I think you alluded to this as well, it's about normalizing the concept of palliative care early on, isn't it? And introducing it no matter what terminology you use. I think the other point is that palliative care is a really scarce resource, isn't it? And I think there's also this kind of, sometimes there's a sort of transition to, oh well, we're referring you to the palliative care, and then it can come across like you're being kind of moved on to a sort of we can't do anything else for you. But I think we probably, as people looking after people living with MSA, also need to upskill ourselves in delivering some of the, because I think there's a difference between palliative care as a specialty and palliative care as an approach, isn't it? And a lot of people who are palliative care specialists would argue that neurologists or whoever is looking after the patients with those conditions ought to be upskilling themselves to deal with some of the aspects themselves. And I don't necessarily disagree with that. I mean, I work with some excellent palliative care people who reach into our clinic, but I think there's an element of that. I don't know what you think, Annette, about that side of things.
Absolutely. Time is often the issue. And I know the Parkinson's UK, they have a module for Parkinson's disease nurses to try and involve them more in that, because they have been more of a reluctance because they don't feel skilled enough in this aspect. I think it's more difficult in MSA because there aren't MSA nurses on the ground everywhere, just because it's not common enough. But perhaps even just giving them written information on what is put in compounds might be helpful.
Developing some of that, sorry. And that is something certainly that we have also had to address and look at. But also I would say that there is a real need out there for, it's great that they get to come and see a consultant, but actually they need someone that they can go to. And one of the things that our nurse specialists often mention is the fact that because it has to be a multidisciplinary approach, there often isn't one single person that people can go to. So therefore, they never quite get the opportunity to sort of think about planning ahead, because quite often they're just dealing with the immediate issue and they don't have time, and they have to go to that particular person to deal with that. And then where next? So sometimes, obviously, sometimes the MSA nurse specialist becomes that person, but actually they can't do that for everyone. They're just not able to.
Perhaps also the other way around is also true, that palliative care is often reluctant to take on people with a condition like MSA that they're not familiar with, because they don't see it very often, and providing training and or information for them to take on these patients would be important. And I have experience of a lot of people who were seen in the general palliative care services in the community, who had excellent care. And I think that came out in the survey as well. Some people do have excellent care in the community, but there is probably a need to training of palliative care service, or this individual palliative care service about the condition.
Thank you. I think we have a question from Professor Kaufman.
I can speak loud. Annette, great presentation. My question is, how does this survey in MSA compares the same items to, let's say, something more benign like Parkinson's disease or ALS? Because without comparisons, it's difficult to see the significance of the complaints.
Yes, that's definitely true. And some of the themes are seen in other conditions as well. So obviously we don't have a direct comparison, but from Parkinson's is probably more comparable than MND, because MND is a slightly different age group and different course. And the carer aspect is very similar. I would say that carers don't get enough or get very little input, and certainly our services are not designed for them. I think the issues are probably similar, except the much more rapid progression, because there are a lot of people with Parkinson's who never get to the severe stage. But I think there are a lot of similarities. I think the ACP planning is probably more important to be done early in MSA, whereas in Parkinson's disease, there's a lot of emphasis on the symptomatic treatment to make people feel better and normal again in the beginning. And then there's often reluctance, or it's more difficult to then approach the ACP discussion. And I think in MSA, there is not the time to do that. But the care of late-stage Parkinsonism and PD-PAL projects, they put together people with late-stage Parkinson's and MSA and PSP because of the similarities.
Okay, thank you. I think we've got time for one more question. I think Professor Panica just wanted to raise a question there.
Question to Annette and to the wider panel. So interesting, 1/3 of the cohort found the information available about MSA to be inadequate. And this is a cohort of patients who are registered with MSA Trust. The MSA Trust has curated a robust and a very thorough set of information leaflets. So, of course, it might be that maybe carers and patients weren't engaging with all the services available to the trust. That could be one side. But imagine if they did. My question is, do you think the way that information is being disseminated to patients needs to be really addressed? Because we have sort of a very sort of a very unidirectional approach. We hand an information leaflet, there may be a 5-minute discussion, they read it. Perhaps does that model have to change from written to, say, digital, or do we have to look at other modes of communication, or do you think an information leaflet is so no longer fit for purpose in the current era?
I think it's probably worth saying that the information is available digitally, isn't it? I think, Karen, you probably can speak to that as well. But I mean, I think the other point about that, I think we raised this when we discussed it at the time, that I think potentially the survey encompasses potentially going back many years, because you have former carers of people with MSA who may not have had access to the current set of information. So I don't think the current needs survey was necessarily addressing the current level of information that's being provided. I think that was what we concluded when we looked at it, wasn't it?
Yes, and obviously I simplified because that was a question broken down in different aspects by provider, and a question was also at the time of diagnosis, and for me, it was really a revelation, not from this survey, but previously, that when we in Parkinson's, when we communicate information at the time of diagnosis, only a small percentage of that actually is heard or understood and retained, because it's obviously an extremely emotional time. So written information at the time of diagnosis is certainly something we should do. And the information from the MSA Trust was very much appreciated. A lot of people find it overwhelming to have all this information. So having someone like the MSA nurse to personalize it, saying this is what's relevant, this is your problem, is also important. Not just saying here it is, find what's relevant to you. These are just some thoughts.
The other issue for us in terms of digital, and we are actually looking at that at the moment, is how do we put it online in such a way that it is easier for people to find it when they particularly need it. So sometimes we sometimes do feel that maybe we've almost overloaded our website because it has so much information on there. And, for some people, that's great. They can be discerning and figure it out. But for others, it was just overwhelming, as you said, and they've just had a diagnosis and they're struggling to actually deal with that. They don't need all the information straight away.
Yeah, I think it very much has to be a personalised approach, doesn't it? So I'm just going to make one more observation before I close the session, because another key need that was mentioned by you, Annette, in the survey was financial information. And for anyone who's looking after people with MSA, the MSA Trust has an excellent team of financial advisors who can help with advice about financial and benefits entitlements. And they've recently hit some key milestones in terms of how many people they've advised. I would really strongly commend them to all people involved in the care of those with MSA, because it's really important for the patients and they really appreciate it. So I'd just like to close out the session. I'd like to thank all the speakers for providing some really stimulating talks, and thanks for some interesting questions as well. And yes, thank you very much.
Welcome back, everyone. Welcome back. It's a great pleasure to start the last but not least session, which is, as you know, from the agenda, based on clinical trials. We have distinguished speakers who will give an overview, an update on some of the latest development in clinical trials. Some of these trials were done in my clinical research facility just around the corner, so I'm particularly pleased to introduce as a first speaker Professor Tom Folteny, who will give an overview, an update on the exenatide trial in MSA. So, Tom, over to you.
Thanks very much, Vincenzo. So I'm going to go quite quickly and just dwell on the important points rather than going into detail about everything. So I'm going to present the trial of using a drug called exenatide for patients with multiple system atrophy. So what is exenatide? It's a GLP-1 receptor agonist that many of you will have heard of because of Ozempic and Wegovy, which is the biggest selling drug around the world for weight loss and diabetes. So in terms of GLP-1, when we all eat a meal, the cells in the large bowel produce GLP-1, gets into the bloodstream, circulates, and hits the GLP-1 receptors on the beta islet cells of the pancreas, making them release insulin. And this keeps your blood sugar under control. GLP-1 disappears within a few minutes because an enzyme called DPP-4 eats it up. However, GLP-1 receptor agonists don't get eaten up by DPP-4, and therefore they work in the longer term, and then they can keep your blood sugar under control in the longer term. That's why they're licensed for treatment for diabetes. There are GLP-1 receptors in the pancreas, good for diabetes. If you stimulate these, not only does it cause insulin release, it also has a trophic effect on the beta islet cells. So it preserves and can lead to increased tissue mass in the beta islet cells. So it's not just an agonist that causes insulin release. There are GLP-1 receptors in muscle, in fat, in the liver, in the stomach, in the heart. And so different specialties are very interested in what GLP-1 receptor stimulation does in different tissues. And of course, we're very interested in them in the brain with regards to neuroprotection. So the first GLP-1 receptor agonist was exenatide, and there have been a number of others. As I say, the market leader is semaglutide, it's most potent, but there's a differential brain penetration between these. So this is work by Vasilios. Just to summarize, the first column of the red blobs shows insulin resistance in brain tissue from MSA patients and PD patients compared with controls. So if you just remember IRS1, the S is the serine phosphorylation. It means you're insulin resistant. So controls don't have much, but there's more in Parkinson's, and there's even more in MSA. And the MAP2 just confirms that these are neurons. And in animal models of MSA, this is the PLP2 animal model of MSA, there's loss of tissues, there's loss of neurons, there's behavioral problems. And in the presence of exenatide, these animals are preserved in terms of behavior and tissue. And together with that preclinical data, back in 2017, we published a paper showing that exenatide in a double-blind placebo-controlled phase 2 trial, there was an advantage in the clinical severity of exenatide compared with placebo. So all of that information made us very keen to see what happens when you use exenatide in MSA patients. Obviously, MSA patients have a more aggressive disease. We had to do an open label trial because we didn't have placebo. As much as we wanted to do a double-blind placebo-controlled trial, we didn't have it, and we couldn't manufacture it because of the costs. It cost millions to manufacture, and the drug company wouldn't give it to us for nothing. So rather than do nothing, we decided to do an open label trial, again, randomized, comparing MSA patients with standard of care compared with standard of care and exenatide. And we decided to use the UMSARS parts 1 and 2 scale because it's the most widely accepted scale for MSA. We wanted to collect pilot data to see what happens. So part 1 is the patient reports, part 2 is the objective rating. Because this was an open label trial, we were really careful to get some very objective measures as well. So the number of people who lost the ability to walk, number of fallers, people who had severe impairments in speech and swallowing, and their global impression were included in our secondary outcome measures. And again, really objectively, we wanted to measure, is there any change in wet biomarkers? For example, NFL, which is the most sensitive measure of active neurodegeneration. Again, digital measures of walking and numerous aspects of walking, and brain imaging. So which patients? We recruited MSA-P and MSA-C, and they were randomized to act either as the standard of care controls or the exenatide arms. And I won't dwell on this, but we wanted just to exclude as few people as possible, that anybody that wouldn't be safe with exenatide was excluded. So we pre-screened people according to their medical records, and then they had a formal screening assessment. We recruited 50, and 25 were randomized to the exenatide arm, 25 to standard of care. 2 of the standard of care people were very disappointed that they weren't going to get access to exenatide, and so they went straight out and managed to access it privately, which of course ruins our experiment. However, we did the intention to treat, which is the formal way, despite that, we looked at intention to treat. We also did a compliance analysis to take into account for those 2 people who had basically crossed over. So our primary outcome was measured at 48 weeks of drug exposure, but then we followed up people for an additional year, so that we have data from 96 weeks when both groups had been off exenatide for the 2nd year. So they were 62 or 63 years old. They'd had symptoms for 4 years, but only had a diagnosis of MSA for about 1 year. Identical gender split, slight excess of women. 14 MSA-P and 11 MSA-C in each of the arms, so 25 versus 25, weighing about 75 kilograms. With UMSARS parts 1 and 2 of 42 in the control group, 46 in the exenatide group. Few people on Parkinsonian medications and medications for orthostatic hypotension, and of course, based on where we are, a vast excess of white British people, but a few ethnic minorities represented as well. So this is the primary outcome. So the top 2 rows are the UMSARS 1 and 2. And you can see, looking across from baseline over to 48 weeks, that there's a worsening of 6 points in the exenatide group and a worsening of 13 points in the control group. So a clear advantage, and that is statistically significant. And so we're interested to know, well, was it the patient reports or was it the objective measure? And so the UMSARS 1, there's an advantage for that, 3 points worsening exenatide, 7 points control. In the UMSARS part 2, 3 points worsening exenatide, 6 points in control. So all of these are statistically significant. So our primary outcome of this result is positive, which is encouraging. We followed up people for the 2nd year, and there was a pretty much complete catch-up. So those people who had been on exenatide, by the end of the 2nd year, were now much closer to the severity of the patients who had been controls. So there's no longer a statistically significant advantage of having been on the drug for 1 year compared to the end result after 2 years. So that argues against there being a disease-modifying effect, because there's nothing that's persistent. There might be a symptomatic effect, but of course there might be a placebo effect or observer bias. So these are the individual scores. Now, I really emphasize the importance of looking at individual scores in small trials to make sure that the data are not being skewed by some individual who's had a massive change or a subgroup. So that generally there doesn't appear to be a particular subgroup. These are the secondary outcomes. So UMSARS part 4 is a global impression, and then there's a CGI as another global impression. Both of those slightly favor exenatide. For loss of walking, there are only a very few people who lost ambulation, so there's no difference there. For speech, there seems to be less people had severe speech problems in exenatide versus control. Doesn't reach significance. For swallowing, again, 10% have severe swallowing problems versus 35% of controls do, and falling 14% versus 26%. But again, at the end of the 96 weeks, all of those advantages have largely disappeared. So the secondary outcomes are not statistically significant, but there's a slight hint of an advantage, which is important to note, but it doesn't tell us much else. Now, at this point, I want to be as objective as possible. So because of the risk of placebo effects and observer bias, we are very keen to look at our wet biomarkers. So neurofilament light you can measure in the spinal fluid or in plasma. And unfortunately, there is absolutely no advantage of using exenatide based on NFL. So the FDA are now approving NFL as a way of looking for neuroprotection across neurodegenerative diseases. So this would argue pretty strongly against there being any benefit of using exenatide in MSA. If anything, there's a small increase in the NFL in plasma in the exenatide group. And we did some very clever work collaborating with Edinburgh, looking at alpha-synuclein oligomers in CSF, and again, nothing was found. The most objective way of measuring gait is using a lumbar sensor. So the activity sensor, people wore for a week at multiple time points, and we could see how much walking they did, how fast they were going, how many steps, how many bouts of walking, lots of different parameters of walking. And again, despite the reports from patients and despite their clinic visits looking perhaps a bit better, there is no advantage in their walking during the course of the 48 weeks of exposure. So again, that is very sobering, that the likelihood of this drug being even symptomatic, let alone disease modifying, is pretty low. And Chris Lambert helped us with MRI scans. So 32 of our 50 patients had baseline scans. Only 14, unfortunately, were able to have MRI scans at the end of 48 weeks. But even with the most lenient thresholds, there was no advantage seen on MRI imaging between the two groups. So, despite a primary outcome being positive, the risks of placebo effects and observer bias is very, very high. We cannot exclude that the drug did have an effect, but when you look at the data in terms of the most objective things, the NFL in CSF and plasma, the activity digital measures and the imaging, there appears to be no robust advantage of using exenatide, which is a big disappointment. Might it help some people? And this is, I suppose, what we're also doing, because in parallel with this, we've completed a phase 3 trial of using the same drug in patients with Parkinson's disease, and we published the results of that. And that trial shows no advantage of using exenatide in Parkinson's patients. So both of these together make the overall interpretation much more negative, shall we say, and much less optimistic. There are trials of using other GLP-1 receptor agonists in neurodegeneration, and there will be some more work done in Parkinson's disease with lexazenatide in France. There are trials of using semaglutide, this is the Ozempic Wegovy weight loss drug, in patients with Alzheimer's disease and in patients with Parkinson's disease, and we'll have to watch to see if those trials are positive. In the meantime, we know that using these drugs reduces the risk of developing things like Parkinson's disease in people who have diabetes. So there seems to be some action for some people, but it might be reserved for those people who have let's say, an insulin-resistant form of Parkinson's or an insulin-resistant form of MSA, not all comers. And so we are doing some work to look at insulin signaling in the brain. I won't dwell on this, but as I said at the start, there's the serine phosphorylation makes you insulin resistant, the tyrosine phosphorylation makes you insulin sensitive. And we can measure that and the downstream effects of that using blood tests. So from the blood tests we collected from patients, we can separate out things called exosomes or extracellular vesicles, and we can find those vesicles which have come from the brain because they have a surface marker that's specific to the brain. And then within those brain-derived vesicles, you can measure to see what has changed over time, and you can look at the insulin signaling pathway. So that is what we are doing at the moment. So although this is disappointing, I think it's important, and I think we've learned a lot from both the Parkinson's and the MSA trials about the role of these drugs in these diseases. So thank you very much to everybody who supported this. The MSA Trust helped patients participate in this by paying for their travel expenses. So thanks to them and for everybody for their assistance. Thank you.
Many thanks, Tom. Our next speaker is Dr. David Stamler from Alterity Therapeutics. And this will be another interesting presentation on different trials, different IMP, different drug. So some results on ATH434. David.
Good afternoon, everyone. Hopefully, can you hear me okay with this microphone? Yeah, thank you to the organizers and to the MSA Trust. This is the first time I've been at this meeting, and I'm quite impressed with the advocacy work that you're doing. So thank you for that. I have the pleasure of telling you about our, oh, wait a sec. I have the pleasure of telling you about our phase 2 trial in MSA. These are our disclosures. Either myself and our other authors are either employees or were compensated for their participation in the study. So we'll be talking about a drug which is targeting labile iron. So I'll give you a little bit of background on that as we go along. Labile iron is required for all important cellular functions, or many important cellular functions, such as energy production. It's required for the activity of about half of the enzymes in the humans. And regarding MSA, iron accumulation is known to occur in MSA-affected regions, such as the putamen, the pallidum, and the nigra. While there's not hard evidence about its role in the pathogenesis, it's clear that this accumulation is indicative of impaired iron trafficking, and that's really what we're hoping to address. Now, labile iron in excess actually causes several deleterious effects in cells. It promotes alpha-synuclein aggregation. It actually promotes oxidative injury, and I'll show you in a subsequent slide. You get this vicious cycle between these two where it really propagates itself. Now, ATH434, I sometimes refer to it just as 434, is an iron chaperone that redistributes this excess labile iron in the central nervous system. It's an oral agent. It is a small molecule, as you can see there, with a molecular weight of about 300. And it's been shown in vitro, in the top panel, to inhibit alpha-synuclein aggregation both in vitro as well as in vivo. In the second panel on the right, you see it robustly reduces oxidative injury that's mediated by an oxidative stressor called menadione. And we've demonstrated efficacy in multiple animal models, both rodent in Parkinson's disease and MSA, as well as this primate model of Parkinson's disease. So I'm often asked, what's the evidence that iron is really involved in the pathogenesis? And I would say that there is a robust amount of data, but most of it is circumstantial. Hopefully our data, which really gets at targeting this iron, will become part of the puzzle. But there's a review article that came out of the Innsbruck group several years ago that summarizes a lot of histopathology data, as well as MRI data supporting this. And then the group from Toronto with Gabor Kovac talks about the close association of iron accumulation with distinct alpha-synuclein pathology in the different subtypes of MSA. So this picture, which comes from the Wenning group's paper, talks about the vicious cycle of cellular injury. So if you recall, labile iron is indicated here in green. In excess, it cycles between the 2+ and the 3+ form in something called the Fenton reaction. In doing so, it generates free radicals, and those free radicals themselves damage intracellular structures such as lipids or DNA or mitochondria. The excess iron itself, as I mentioned, promotes this alpha-synuclein aggregation to form these oligomers that then form aggregates. And then this table on the right, I won't dwell on this, but it really talks about what excess labile iron does, and then how the aggregating synuclein itself promotes neuronal dysfunction. There's a recent paper on this out of New Zealand and Australia. But the aggregating synuclein actually promotes further free radical production. So what we're really trying to do is, by getting rid of this excess iron from the cell, to reduce the drivers of toxicity and to break this cycle. So to kind of briefly summarize, we aim to redistribute the excess labile iron, in doing so to reduce synuclein aggregation and oxidative injury, thereby preserving neurons and oligodendroglial cells, and then hopefully ultimately stabilizing or improving function. So our objectives of the study were to evaluate the efficacy, biomarker response, and safety of ATH434 in patients with MSA. So we're aiming to randomize 60 patients to one of 3 dose groups in equal amounts, 75 twice a day, 50 twice a day, or matching placebo with 12 months treatment. The diagnosis of MSA, which everyone has fully appreciates, is quite difficult. So we did use the revised established MDS criteria. We also required elevated NFL at baseline, and we did also require evidence of increased iron in a basal ganglia area, as well as relatively short duration of motor symptoms and no evidence for severe impairment. I won't talk about this in detail, but our collaborators at Vanderbilt have really driven the neuroimaging component of this trial, and I think they've really helped us quantify iron, and we also use this for qualifying patients to come into the trial itself. So the visit schedules are here. We conducted an MRI at baseline 6 months and 12 months. We conducted our clinical exams including the UMSARS and a clinical global orthostatic hypotension symptom assessment and wearable sensors every 3 months, and then safety visits were done frequently. As a footnote, we did collect both plasma and CSF NFL. There were bioanalytical issues with CSF NFL. That's still ongoing, so I don't have that to disclose today, but we will be reporting that in the future. So we wound up randomizing actually 77 patients, not the planned 60. And we excluded from the primary analysis, which is the imaging analysis population, 16 patients. So 5 did not have aggregating synuclein of either the MSA phenotype or the PD phenotype. 10 discontinued before week 26, and 1 completed but the week 26 but could not conduct an MRI. So that left us with 61 patients. The primary endpoint in the study is change in iron content in the Niagara by MRI. No one's tried to do this before, and we figured given our mechanism of action, we would kind of aim for this. So that's why this population that we're targeting is we're referred to as the imaging population. The criteria to be included was at least 1 post-baseline assessment, as well as a positive aggregating synuclein assay. So we did explore a secondary population given the large number of patients who were not included in the imaging population, and that we're referring to as the clinical analysis population, where we basically required patients to be randomized, treated, and have 1 post-baseline assessment of the UMSARS, which was at 13 weeks, and that gave us 71 patients. And these are the patients that I'll report for the remainder of my talk. So this was the pre-specified key secondary endpoint, was the change in the modified UMSARS-1. This modified UMSARS-1 uses the standard scale but excludes the sexual function item given its historical non-responsiveness. So these are the baseline characteristics for the MITT. Typical for an MSA population, age in the low 60s, slight male predominance. The modified UMSARS score was about 15 to 16, and you can see the plasma NFL is in the low 30s. Now, one important thing to note is the prevalence of severe orthostatic hypotension at baseline, which I think most people appreciate as a predictor of rapid disease progression. We only had about 1 patient in both the placebo and the 50 milligram dose group, but we had fully 33% or a third had rapid progression, or sorry, had severe orthostatic hypotension, which is defined as a drop from sitting to standing for 3 minutes of 30 millimeters of mercury or more in systolic blood pressure. So this is the key clinical endpoint. I will come back to the primary endpoint of iron, which I know will be of interest to some people. So this is the UMSARS-1. Obviously, higher scores are worse, and we see that in the placebo group, there was an increase of about 8 points over the span of 52 weeks, whereas in the active dose groups, there was considerably less deterioration. The placebo, if you look at the difference between active dose group, 50 milligrams, there was a 3.8 point difference with a relative treatment effect compared to the change in the placebo of 48%. And at the 75 milligram dose group, it was lower at 2.4 with a relative treatment effect of 30%. Now, we've been analyzing the data, and I don't have any definitive answers as to why there's a lack of a dose response. Our statistician, who's very seasoned, says he doesn't necessarily believe there is, given the relatively small sample size, but we're continuing to investigate. What I will say is that we have just conducted an informal analysis of using the severe orthostatic hypotension as a covariate, the actual change from sitting to standing, and that did actually diminish this difference between the groups by about a half point. So I think we're getting closer to the answer. So this is a clinical global impression of severity, a 7-point Likert scale, where higher scores are worse, and we see the placebo patients over the span of 52 weeks increased by 1.2 points. The 50 milligram dose group, in keeping with the UMSARS data, deteriorated less than a half point, and the 75 milligram dose group was somewhere in between. Regarding the orthostatic hypotension symptom assessment, this is the change from baseline to week 52. These patients started with a baseline score of about 13 to 14 on this scale. And over the span of a year, they deteriorated by about 6 points, whereas there was improvement by about a half to 1.5 points in the active dose groups. And these differences were not statistically significant, but were approaching some. Now, these, I think, are interesting data. We did use wearable sensors that were validated for use in the clinic. And patients wore a pendant around their neck for 2 weeks at baseline, then in every 3 months afterwards. And what you see in these different parameters, let's focus on step count, that in the placebo patients, there was about a 2,500 steps per day loss in the placebo groups, whereas in the active dose groups, we saw about half that loss. So it really does appear to be preserving activity in the outpatient setting. If you look at total walking time, bouts of walking, this is just getting up from a chair and initiating an episode of walking, as well as total standing time. So regarding adverse events, there were similar rates of adverse events across the dose group. The most frequent adverse events are listed here: UTI, fall, COVID-19, et cetera. No evidence for increased frequency of adverse events in the active dose groups. Similarly, no signal for severe adverse event incidents, and there's no evidence for increased serious adverse events. None of the SAEs in the study were attributed to either study medication, which is placebo, or 434. Now, regarding change in iron content, as mentioned, this was the primary endpoint for the study. We did have a certain understanding of the drug before we started the trial. That understanding did change during the course of the trial. But what we did see was that in both the 50 milligram dose group and the 75 milligram dose group, that there were reductions in iron content in the pallidum across both dose groups, to a lesser extent in the putamen. And then, interestingly, in the NIGRA, which was the primary endpoint, only in the 50 milligram dose group. I think there are technical reasons why we may not have been able to see this in the NIGRA because of the small size of the region, as well as trying to register an MRI both at baseline and at 52 weeks. Nonetheless, we do believe that there's evidence for reduced or stabilized iron content in the pallidum and as well as the putamen. And we did see reductions in the 50 milligram dose group that was, but not at 75, which was the primary endpoint. Now, we also did, our colleagues at Vanderbilt also looked at the group change, where you basically create an average patient at both, where you combine all the patients in a dose group, and then you look at that change in their iron content over 52 weeks. And that gets away from this registering scans at baseline and at end treatment. And then this is basically the change in the iron content in the putamen and in the pallidum. And you see hotter colors or increases are red to yellow. And what you see is in the placebo, a fair amount of red and yellow, indicating increases in iron over the 12 months of treatment. Whereas in the active dose groups, you don't see that yellow at 50 milligrams, and then 75 milligrams is somewhere in between. So in this group-wise analysis, we see increases in placebo greater than iron, and again, some evidence for target engagement in the globus. So this I found interesting. I just want to say we didn't include standard error bars because they're quite large here, but this does show the change in brain atrophy in something called the MSA Atrophy Index that the folks at Vanderbilt developed, which is a composite Z-score that looks at both the putamen, or looks at the putamen, globus, cerebellum, as well as the brainstem. And you do see here again negative changes indicating atrophy or worse, and you do see decline in placebo that is smaller at 26 weeks, and it appears to that difference appears to be growing over time. So I think this is intriguing. This is using MRI to try and effectively look at neuropreservation. So it's a rather crude tool, but I think it's suggestive that we may be doing something beneficial. So in summary, ATH434 showed clinically significant efficacy in modifying disease progression based on the UMSARS-1, as well as based on other secondary outcomes. There were baseline differences in disease severity that likely explain the differences between 50 and 75 milligrams. We did demonstrate target engagement by reducing iron in areas of the brain that are affected in MSA. We did see what I thought was a favorable safety profile with similar adverse event rates. We don't have time to talk about the alpha-synuclein data, but we did see, as I think Vasilios mentioned, patients in this trial that clearly had MSA that either had a Parkinsonian variant or a mixed phenotype. So that is definitely a work in progress. And I think the results support continued exploration of targeting labile iron for treatment of certain neurodegenerative diseases. These are our investigators. Many are here in the audience, so I really want to thank them for all their hard work, their coordinators, as well as the patients who contributed so much to the study. So thank you for your attention.